presentations from Dr. Aidan on the animal science, the greenhouse gas emissions, how methane is produced, and the life cycle that it goes through. Then there are some interesting presentations from the Global Methane Hub. One question that stood out was how do we get continuous funding to look at how we can monitor methane? The response from the Global Methane Hub resource person was that, well, they do a lot of ecosystem events, putting together people, and they recommend for such grants or activities to take place. So I'm sure there are avenues for collaboration there that can be explored from that point of view. And then we had Dr. Odami and Dr. Selassie taking us through the current trends when it comes to materials for capturing carbon, and then how AI and IoTs are helping to do rapid determination or measurement of carbon, and then how to make data available. Some of the presentations were interesting, as open as possible. It was also built on some of the things that those of us here made, the issues for making data openly available, the issues of artificial intelligence, why we have to take very cognizant notice of it, not to trust whatever it gives us, and so on. Today, we can think of it like yesterday was for those on the project. Today's program is for current researchers on the program. We take an update from Germany, and then I'll call it current researchers and next generation researchers. They are coming to tell us some of the interesting things that they are doing, and it promises to be exciting. Without taking too much time, because time is fast spent, I'll invite Dr. Jeroen Henricus Theodor Zethoff, I hope I got the name right, to give us an update from Germany. Can we give him hands of applause as he comes to Mount Video?
Presentation coming on screen. It's standing here.
Okay, let me tell them.
Yeah, super. It's good, right? Readable? Yeah, right. So, yeah, thanks a lot for, yeah, it's working, thanks. Yeah, for the opportunity to present here our first analysis. A bit more higher. The tall Dutchman, yeah. So I will present here the data that we got from our partners in Tingvoll, from Noordzok. They collected their data since 1991 when they established this farm, and yeah, I analyzed it. And so to take you now more north. Is this working like this? Okay, it's in the middle of Norway, so quite north compared to here. And they established there an organic milk production in 1989. They use conventional grass clover as fodder production, and these fields, they have to be plowed every 4 to 5 years in order to reset the grass clover succession. Otherwise, you end up with only grass and weeds. Since 2010, they also started an additional experiment at this farm, where they installed a biogas reactor, and they were using anaerobically, or as a byproduct, they have this anaerobically digested slurry, and they are now looking at if this can still be used as a fertilizer, or if this is producing only extra greenhouse gases. Yeah, so as said, they started in 1991 with monitoring, and you see here in red the mean annual air temperature, but from the 1st of April to the 31st of October, because during the winter time they cannot really operate the weather station, So we cut down to only the growing season, more or less. And then we see clearly a climate change effect because there's a constant increase in temperature over the growing season. There's also a small trend in the precipitation. So the prediction is that this area of Norway will become wetter. We see this slightly, but it's not a very clear trend. In the background, you see plotted the yield of the grass clover lay per year, whereby they normally have 2 cuts, but you see that since 2012, they start to have a 3rd cut. This is in part due to the lengthening of the growing season. So again, you see the climate change effect. And maybe if I take this plot, you see it clearer that over this, this study time period, you see that the yields are declining. So while we have a 3rd cut, we have a longer growing season, the overall yield is declining, especially for the the 1st cut. You see this, the dark dots. So in this presentation, I have basically 2 aims. We want to assess if this decline in yield is related to soil organic matter content and a nutrient station. Nutrient status. And secondly, I will talk a little bit about this digested slurry, how this is maybe a way to deal with soil organic matter content and quality, but Johanna will focus later on more on this. So as said, they started with sampling in 1991. They took samples actually till 30 centimeters depth, so these are very shallow soils, but they couldn't even get that deep at all the spots, so I focused here only on the top 20 centimeters. What you can see here is that we see a clear decrease since that they converted the farm to an organically managed cow dairy farm. And this is in partly because they restored the drainage system in the fields, and they also applied a lot of lime, as the soil was quite acid. It's not only between 1991 and 1995 that we see a significant decrease, but especially in the latter part. So you see that 2015 and 2021 were significantly lower than 1995. So this decrease is still ongoing. So they also measured, they measured total nitrogen only in 2009 and 1995. So that's a bit of pity because, they use grass clover, and clover is fixing nitrogen. But most interesting here, what you see is that the middle one, so the available phosphorus, is declining since they start monitoring. And these are averages, but some fields are even below advisable levels for grass clover lays. And I brought this all together with the principal component analysis. And what you see here is, the main message here, so to say, is that you see that soil organic matter is closely related to phosphorus availability. So it seems that the decline in soil organic matter might be related to a lack of phosphorus application, which has to do, of course, with their organically management, where they want to apply as least fertilizers as possible. Also, if we're looking at the farm gate nutrient budget, so this is a budget, not a balance, to be clear, because they don't have any data on leakages or greenhouse gases emissions. So, but here we see that in the dotted line, that phosphorus was basically not applied till 2005. Then they start to get in some phosphorus, but it's still, the decline seems to start to level off, but it's not enough still, so to say. On the other hand, we see that potassium and nitrogen don't see any issue here at this farm. So as said, they installed this biogas reactor, and they set up an experiment where they have this digested slurry and just normal slurry, cow slurry, and they applied in the same amount of nitrogen content. I should add that when you digest this slurry, that you basically lose mainly the carbon components. So if you want to apply the same amount of nitrogen, you use basically less of the anaerobically digested slurry. Another important thing is that when you digest the slurry, it becomes more liquid and also less hydrophobic. So it's infiltrating deeper into the soil. And Johanna sampled in 2024. And she sampled over basically 3 depths, so 0 to 5, 5 to 10, and 10 to 20. Again, the soil is very shallow, so much more deeper we cannot get there. And interesting first is, of course, that you see an increase in blue is the anaerobic digested slurry. And you see that this increase is higher after 14 years of application than business as usual, which is green, or the control without application. The digested slurry even led to a significant increase in carbon and nitrogen content compared to the control after 14 years. So this is also a long-term effect, I want to stress. But if we are looking at the soil organic carbon stocks, because in the beginning I was talking about that we see a decline in soil organic matter at this farm. We see the same for this experiment, that in 2011, when they first sampled the carbon, it was higher than now. But more interesting is that the decline is least for the anaerobically digested slurry. So Johanna will go into more detail there later on this morning. But this is, I think, quite interesting result that we already found. Especially considering that, as said, with the digested slurry you apply less, so there's less carbon coming in. We have to look at the yield data, so maybe it might be that there is a better plant growth. So to summarize, here on our Norway side, we see that soil organic matter is declining, partly due to these nutrient imbalances. But we will focus with further incubation studies on what this actually means for the carbon and nitrogen dynamics in the soil, especially if this digested slurry might be a solution to limit carbon losses from the soil. So thank you for your attention and questions. Working.
Good.
Hello.
Yeah.
Hello.
Great.
Jeroen, thank you very much. The interesting relationship between soil organic matter and phosphorus availability. A lot of us don't really focus so much on phosphorus, but have you got any indication of the mechanisms? How does that process work in terms of, is it a microbial process that uses phosphorus to create organic matter, or?
Sorry, to create organic matter from phosphorus?
Is it a microbial process that causes the organic matter to increase in relation to phosphorus? What do you think the process is?
I think it's mainly about phosphor mining then. So if phosphorus, because I showed phosphor availability, by the way, and the general theory is that when phosphorus is limiting for plants, they stimulate mycorrhiza, mycorrhiza, and that they are able to basically, get the phosphorus available for the plants from the soil. Something also what we want to measure is what's actually the phosphor stock at the site. So availability is one thing, but stock is of course the second thing. How much? But at least it's very clear also because from what they were telling about to us about the practice that they really focused on lowering how to say, fertilizer input, because for these, in Norway, the biggest issue is that the fjords get eutrophied, so there's a lot of washing of nutrients into the fjords, which cause a lot of ecological harm.
To add to what Jeroen said, even within the African system, we have seen almost a similar trend, where what organic matter do, especially the functional group, they are also negatively charged. So they compete with the phosphorus for suction sites, making them available in soil solution. So we found certain trends linking to what we found in Norway.
Okay. Oh, nice. Interesting. So we should compare at some point. Yeah, nice. Thank you. More questions then?
We have seen cases where carbon stock seems to decline in agroecosystems despite intensive organic matter management. And that has been an issue for discussion. And I remember in our first EJP, one of our first EJP meeting, this issue popped up and Abad made some very nice contribution, which may likely cause this decline. I think that, I believe this is what you are noticing here in terms of your soil organic matter decline. I mean, what could be some of these reasons
of the decline?
in your observation?
So the decline is in part I think the nutrient imbalance, and in part of the liming effect, so to say. So they apply quite some lime. In the beginning, they used shell sand, so with calcium carbonate, which is very easily available and which is really increasing the pH and let the microbes go crazy, basically. And they continue to keep the pH on quite a high level for these climates. So that might also be one of the reasons why the soil organic matter is declining. Yeah. So and the draining of the soil. So some of the sites are not that long in use. So I'm talking then about 50 years or so. And previously it was like a forested site. So it might be that there's still some background organic matter because the soil is quite rich in soil organic, or it was very rich in soil organic matter, I should say, in the beginning. So there are basically three main reasons why the soil organic matter might be declining for our site. And maybe that's here as well, I don't know. Yeah. So thank you.
We have some 2 or so minutes. If there are any questions, you can take it. If not, excuse Dr. Jeroen for his interesting presentation. You know you are next. The presentation. Are you part of the carbon dioxide African market? Yes, what is it? It's not yet your turn. It's not yet your turn.
Oh, it's not my turn. I
Yeah.
thought I'm directly after.
Don't worry. We'll mix it up.
Now it's.
Okay, so you are. No. So you are.
Miss Johanna Zimmerman.
Thank you. So next is Miss Johanna to take us through nutrient and carbon dynamics of different land use systems. Thank you. So 15 minutes. And then
Yes.
use this mic too so we can hear.
Oh, not this one?
This is a little bit clearer.
So, good morning, everyone. Today I give you a short overview of the experiment that I'm working on. And like I said yesterday, this research is part of a collaboration between the RPTU in Germany and Norshock in Norway. So first, I would like to present to you our team. Our team consists of 5 people, 4 based in Germany and 1 in Norway. So Professor Dr. Hermann Jungkunst, Dr. Jürgen Zethoff, who's sitting over there, Dr. Klaus Schützenmeister and I are working from Germany, and Dr. Tatjana Rittel is our partner in Norway. So this experiment takes place at an organic research farm in Tingvoll, Western Norway, and was established 2011. The farm includes 22 dairy cows and a biogas plant, and the field is managed as a grass clover lay for 4 years, followed by 1 year with another crop, for example, oats or peas. We compare anaerobic digested slurry from the biogas plant with untreated slurry, both applied at 220 kilograms of nitrogen per hectare per year. And in that 5th year, in which we have another crop, the field is plowed and we have no application of the slurry. The experiment is set up as a randomized block design, as you can see on the picture. So to give you an overview of the timeline of our field activities, we started sampling in April 2024 with the first soil and plant sampling, and we started the gas measurements. In May 2024, the field was plowed and some oats were sowed. So you can see we are in the 5th year of the crop rotation. Then during the summer, a master's student of mine continued the gas measurements. And then the oats were harvested, and she took another soil and plant sampling. In August 2024, the grass and clover seeds were sowed to have the first year of the new crop rotation. And in May this year, there was the first slurry application, and in October 2025, I was again there to take a 3rd soil sampling. We took the soil samples from 0 to 5, 5 to 10, and 10 to 20 centimeters. Like we already discussed, the soil is really shallow, and after 20, maybe 30 centimeters, you reach the bedrock. And they had some soil sampling from 20 to 40 centimeters, I think in 2011, but it was made with some kind of a device, so it's not possible by hand. And we measured pH, soil texture, bulk density, soil organic carbon and nitrogen stocks, and I also did some measurements about soil organic matter stability. So I did a thermal analysis of soil organic carbon, and a bachelor student of mine did a cold and hot water extraction of carbon and nitrogen. And we also took some samples to assess the extracellular enzyme activity, which were then measured in France by Abad. So to give you some first results or some key results, for this, please keep in mind we are in the 5th year, so we have no slurry application. So if I'm speaking of an effect of the type of the slurry, it's more like a residual effect and not a direct effect of the application. So we observed higher carbon dioxide and lower N2O emissions from the plots fertilized with anaerobic digested slurry during the summer and autumn. And we also have higher soil organic carbon and nitrogen stocks in the ADS plots. For that, I need to mention we are applying the same amount of nitrogen, but we are not applying the same amount of carbon. So the plots which were fertilized with the anaerobic digested slurry conceiving less carbon because there's less carbon after the digestion still in the slurry. So even though we're applying less carbon, we have more soil organic carbon in these plots. And it's increasing by depth, so the highest stalks are in the 10 to 20 centimeters. As I said, I also did a thermal analysis, and in the 0 to 5 centimeter layer, we observed higher proportion of stable carbon in the anaerobic digested slurry fertilized plots compared to the untreated slots, but less in the depths of 5 to 10, and in the depth of 10 to 20, it was pretty much the same. And as I said, here are the results from the enzyme activity. So we have higher activity in the plot in the 0 to 5 centimeter layers in the plots were fertilized with the untreated slurry, but in the 10 to 20 centimeters is the other way around, but we have a higher variation in the ADS treated plots. So to give you some ideas what we are planning next, like I already mentioned yesterday, in the last field campaign, I took 200 kilograms of soil with me to set up a column experiment during the winter to measure processes in the soil during and after the application of the two slurry types. And we also compare two different precipitation scenarios in regard to climate change. So we have a historical precipitation scenario, and we also have a modeled future scenario with more precipitation. And Jeroen will also do an experiment about the vulnerability of organic carbon against decomposition. I did something on the computer, I'm sorry. Okay, so we also have samples for that from 0 to 20 centimeters. So in spring, I would like to do a fourth field campaign, and I would like to take another soil sampling, and I would like to measure greenhouse gas emissions before and after the slurry application. And we are also working right now at a manuscript about, for a method paper to find some indicators of soil organic carbon persistence. And we hope in summer we can submit a paper about the field data that I presented before. So thank you for your attention. Do you have any questions?
A little question, maybe it could be stem from my inability to follow promptly, but in your decomposition study, the laboratory incubation, I think I received information that you were going to start it in 3 months ago. Any updates on this very important incubation work? What are preliminary observations?
I'm not quite sure which incubation experiment you mean.
The vulnerability of carbon to decomposition.
That's planned for the next month.
Oh,
so
next
we didn't
month.
do that yet.
Oh, okay. All right. Okay. So I will only request that you follow the protocol in terms of frequencies. Yeah.
Yes. But I think we need to discuss. So our protocol, there are a lot of measures, and I think we need to discuss which one are must-have and which one are just nice to have, because there are so many parameters that we agreed on?
Yeah, in fact, I think the major focus is on carbon dioxide, but for Emmanuel and Joshua, for instance, they are measuring 3 gases because we export ours to Kenya, a Kenyan laboratory, and they cannot run only carbon dioxide for you using the infrared. So they run for methane, nitrous oxide, and also for carbon dioxide.
Yes, we also measured methane and ammonia, but the results were not that interesting, so that's why I didn't show it.
Oh, okay. Yes, I'm done, yes.
Yeah, thanks very much for educating us. We've learned very much following the presentation. I would like to understand the science behind the figures with the CO2 emissions and the nitrous oxide emissions, because you are operating from a different climatic environment, from the temperate environment. We want to understand what is driving the emissions. Yeah, so if you can move to one of the slides where you show the CO2 and then the nitrous oxide emissions as a result of the amendment. Yeah, sorry, I need, at least this should be fine. Yes, so we want to understand the science behind the figures, if you can help us, so that we are able to compare it with our own data from the tropical environment.
Yeah, we are still discussing about that ourselves, because for the CO2 emissions, I hypothesized that for the anaerobic digested slurry plots and the untreated slurry plots, I thought it's the other way around. So it's not that big difference between them, but I thought because if we are applying anaerobic digested slurry, we have less carbon and we have more stable carbon in it. So I thought when I apply that, there will be less carbon dioxide emissions. So we still need to discuss that because it's not that what we were expected. And for the nitrous oxide emissions, there were some studies where they measured immediately after the application, and then for the anaerobic digested slurry plots, the N2O emissions were higher than from the untreated slurry plots. So maybe directly after the application, there will be more N2O emissions, and then a year after, like we measured, there's already some gone, and so we have less emissions. So to make a statement for that, we should do more gas measurements. That's why I want to measure directly after the application next year. And for the control, you can see that the control have more N2O emissions. I have the daily measurements, and you can see during the summer the control has more N2O emissions, and we hypothesized because the plots of the control has less vegetation on, so the soil temperature is higher. So we hypothesized that because we have higher soil temperature, we have more N2O emissions during the summer from the control plot.
It's quite interesting. Because of our interest, we will follow up with personal discussions. Thank you very much.
Thank you.
Any small update on the cumulative emissions?
Oh, there. Sorry. Yes?
Emissions, because I see your values being even higher than in average values reported for tropical soils. So I was wondering, I was just asking, in terms of the tropical ecosystem and the temperate regions, which one is more likely to emit more of the CO2?
You mean if the field in Norway or a tropical field is emitting more?
Let me help Emmanuel. Emmanuel, it depends on management. If you look at her work, for instance, it's under organic amendment, which is slurry from digestate being used to, fertilize your soil. So if these are organic amendments which are freshly applied, you're going to see more emissions than in natural ecosystems where you don't have any application of organic amendment. So that should guide you in your questioning. Thank you very much.
Yeah.
Any
Thank you.
Hello. Johanna, I think part of what Joshua is and
Emmanuel
asking is probably you compare your methodologies in the calculation of the cumulative carbon, like the carbon dioxide and the other greenhouses. So whilst this is going on, I think Emmanuel will need to see you.
Yeah, we can compare our measurements, right?
Yes. Okay. But it's cool for them.
Please, let's do that. Thank you.
Thank you very much, Joanna. Caleb, please. Please, let's give it up for her once again. Okay, so thank you. Up is Joshua Abisa, who will talk on soil, carbon stocks, and nutrient characterization of different land use systems of long-term experiments in Norway. Thank you.
Good morning, supervisors and colleagues around. I'm here to present on soil carbon stocks and nutrient characteristics of different land use systems of long-term experiment in Norway. So my Norwegian sister has come to present on Norsok. So I'm also presenting from Nibio. Okay, so as a form of introduction, soil serves as a key reservoir when it comes to soil organic carbon. However, it's also vulnerable when it comes to the climate change that is across the globe that everybody is talking about of late. And so soil organic carbon also serves as a key indicator when it comes to talking about soil fertility and also soil health. And also play a key role in the soil ecosystem. Now, when it comes to the maintenance or the management of soil organic carbon, there are three key pathways that we look at. One of them is the addition, another is the reduction or trying to reduce the losses, and also protecting the existing quantity or the concentrations that we have in the soil. Therefore, this mostly is more noticeable when it comes to the monitoring, because soil organic carbon, effect or the noticing effect of it is not immediate. It needs a long-term experiment for this to be noticed. And so with this work, it's also good to also monitor the particulate organic matter and also the mineral-associated organic matter to be able to tell if you are protecting the existing carbon, is it efficient or not. However, there is little attention when it comes to the maintenance of soil organic carbon. Though there is deforestation going on, exhaustive cropping system, we don't pay much attention to the maintenance of this. So in most cases, you realize that the soil organic carbon across the land use systems are declining. So when you look at this field I'm working on, it was established in 1988, but it was fostered in 1935, where the land was deforested, and then it was used for pasture, and along the line, it was used for this research station. And also, this needs that is because of the little attention to the maintenance of soil organic carbon, there is interference when it comes to the long-term monitoring or build-up of this soil organic carbon in our ecosystems. And also there is limited data when it comes to the interaction between the land use type and our regenerative practices of soil organic carbon in the natural and agro ecosystem. Therefore, there is a need for a robust management of this, of our crops. When it comes to crop production, there is a need for a robust management that is able to improve the productivity and at the same time reducing emission. Yesterday, one of the speeches we had here, one question that came out was, okay, if a regenerative practice is increasing yield, what is its relation when it comes to the emission? We don't have to practice something that will give us high yield, but at the end of the day will give us more emission. We should have something that will be at par, so that if the production is increasing because of the begonin population, we can also reduce when it comes to emission of the carbon dioxide, methane, and then nitrous oxide. Therefore, the main objective of this research is to improve crop productivity and also environmental quality, as I mentioned earlier, and this can be achieved through soil carbon sequestration. But specifically, we want to evaluate the soil organic carbon pools and the selected land use system in the long-term experimental site in Nibio, and also measure the vulnerability of organic carbon to decomposition from the samples taken. And the third one is to assess the impact of crop diversification on the soil nutrient concentrations or dynamics. And the last, we know we are an agric system, we can't do without crop yield. So that would be the fourth specific objective to look at. And so the hypothesis that we are testing, the first one is to see if carbon stabilized in POC and then MAOC pools will vary considerably among the land use systems in the long-term experiment due to crop diversification and or management. So these are the hypotheses that we're testing in this work. Okay, so to be able to achieve this, the field experiment, as I mentioned yesterday, those of you who were around, was carried out at Nibio, the site, and these were the other information that I provide here. And as I stated here yesterday, so since the screen is not showing, I can't explain it here. So we have 12 land use systems, but when I go there, because of time and other logistics, we're able to sample 10 out of these 12 land use systems that were used for my analysis. Okay, so the table 1 indicates the structure or the features of the system that we are talking about here. So when we take the DAI, which is purely a fodder production, and this DAI consists of the wheat. We have a 3-year-old grass and 2-year-old grass and 1-year-old grass. The reason we have these years attached with it is that on the field, these crops are rotated every 4 years. So at every 4 years, these crops are switched. Where the first one is, it moves to another one, then the second one also moves to the third one in that order. That is why we have the 3rd-year grass being displayed here. And the tillage is mostly plowed. That's the tillage practice that is being implemented over there. And yes, there is a manual production application there. At the same time, there is mineral fertilizer application. So in a form of ISFM practice over here. So as it's been displayed, that is how it's shown on the field. And as we know from the EJP protocol, we sample from 0 to 10, 10 to 20. And as I stated yesterday, because of the nature of the land, we couldn't go to the 100 centimeters. So we ended at the 50 centimeters. And as I stated yesterday, the samples, how they were dried or prepared. And so the data obtained were analyzed using the R statistical software. And so the first objective was to evaluate soil carbon pools and then under this selected land use system. And so our first results shows the POC, the particulate organic carbon. And we see that the eco, which is ecological system, as I indicated in the first table, is showing the 2nd-year grass is having high POC compared to the rest, which means that it's significantly higher when you compare to especially the reference system. The reference system is a system that we don't have cover crops on it. When we compare to the DAI, which is a fodder production, we have cover for the wheat that was there, but we don't have that for the grasses. And then when you compare that to the OPT, it's also similar there. We have cover for them. Now, when it comes to the DAI, which is the fodder production, the MAOC is having higher MAOC at the DAI 3rd year, which I stated yesterday that it is mostly good to have these 2 pools in your soil. But a state where you have more of the MAOC, then it means that is a good way for sequestering carbon for a long time. Because if you have more POC, it means that you are going to have within 10 years or less, you are going to lose all your soil carbon to the atmosphere because of plant uptake and the rest. And so, as I stated again yesterday, when you look at the DAI, for example, the third year, second year, and then the first year, the MAOC increases by the years. So the first year is lower compared to the second year and in that order, which means that the third year MAOC is significantly higher than the second year, and the second year is also higher than the first year. And so the POC for the 0 to 10 centimeters also follows similar trend, where we have the eco second year having more a significantly higher POC compared to the other systems. Now, when it comes to the MAOC2, similar trend follow from the 0 to 10 centimeter depth to the 10 to 20 centimeter depth, where the DAI, which is the fodder system or fodder production system, the third year grass in it having more or significantly higher MAOC concentration than the other systems. And then the lower depth, which is the 20 to 30, the system, as I indicated, the eco, which is the ecological system, is having similar from the 0 to 10, 10 to 20, and then the 20 to 30. And similar trend is seen in the MAOC 20 to 30 centimeter depth. Now, when it comes to the stabilization, it's also clear here that there is about 60 to 81% of the MAOC is contributing to the total organic carbon, which means that it's very high. None of them is below 50. The least was around 62%, which is a very good thing for the Norwegian side, that we have more of the carbon being stored in the mineral associated pool, which means that it's not easily, it's not going to be, when it comes to the release of it or the loss of carbon, it's not going to be that frequent. Though because of change of climate and the rest management, it may change, but since we have more in the mineral associated, then it's a good news for all of us. Same trend applies when it comes to the 0 to 10 depth, 10 to 20, and also the 20 to 30. And also, when you look at the soil carbon stocks and then the specific surface area, as I mentioned yesterday, okay, it shows that where you have high specific surface area, you also have high soil or carbon stock over here. We see that summary at the end of this slide, and that continues from the 0 to 10, 10 to 20, and 20 to 30. And so the vulnerability also indicates that for the first day, almost all the treatment, all the plots were having high emission. But as the days goes by, the emission begin to decline. That is what was observed for both systems. I'm told I have 3 minutes, so let me rush through. So with the objective 3, which is to assess the impact of crop rotation and diversification on the box soil carbon and nutrient concentration, those ones too, we have the total carbon also indicating very good at the 0 to 10, and then 10 to 20 as well. Then phosphorus also indicate here being higher at the OPT, which is the optimized system than the reference system, the fodder system, and then the carbon system. with the grain yield also, it's also indicated here that the grain yield that we obtain, it is the oats that produce the highest when you compare, or significantly higher when you compare to the grain yield. But with the grasses, it is the third year system, which is under the fodder system that produce significantly higher compared to the rest. Now, with the correlation here, as I indicated earlier, when you look at the soil carbon stock with the specific soil surface specific area, it is showing that it is having about 82%, which means that soil carbon stock is highly correlated when it comes to soil surface area. So the higher the surface area, the higher the soil carbon stock that we observe here. And also the clay content is also negative, but then not highly correlated. So based on these results, I can conclude by saying that the optimized system, especially the plot that had the barley and the cover crop, hold the promise for increasing MAOC in the long-term experiment, and also the carbon dioxide flux decreased sharply till the 7th day. So when you look at the table as shown, you see that for the first day it was high, but as the days goes by, it begins to decrease. I mentioned yesterday that we don't have the full results. We are waiting for the Kenyan people to send us the last result to add it to it. So because of time, I can recommend that the optimized system, which is the barley plus the cover crop system, recommend for optimized system is recommended to increase soil carbon persistence. And also the barley cropping integrated with surface harrow and no tillage is recommended. And also I could recommend the ISFM, which is integrated soil fertility management, for recommendation, and also for our use wherever we want to increase crop soil organic carbon. I want to use this opportunity to thank EJP and then my Norwegian mother as well for their support and everything. In this case, I want to say Tak Ali Sama, which means thank you all.
Thank you very much, Joshua, for the interesting presentation. At some point, I thought Joshua was done with his PhD, then he says he's still waiting for results. I'm like, the results you have. Thank you. We'll now open the floor for questions. Yes, Joanna, let me give you the mic.
Thank you for your presentation. It's nice to see some results from Norway too. I have a question to your results. So you have your different managements on the x-axis, and you have four results for each field. So is this from different years?
Different years?
Because you have your management, and then you presented, for example, carbon content for every crop rotation.
Yeah.
Are these data from different years, or do you have different fields there?
Oh, they are the same field, but then each system has 4 different plots in them. So each, when you pick the BAI, which is fodder production, so when you go to fodder production, we have wheat with cover crops, we have 3rd year grass, we have 2nd year grass, and then we have 1st year, forming the DAI.
So you have different crop rotation on the same field. Okay,
Yes,
thank you.
yes. But they are rotated every 4 years.
Yeah, sure.
Yeah.
Joshua. Thank you, Joshua, for your presentation. I wish to see that mother of yours.
I had wanted to show her photo, but because of time,
No, that's good.
She's called Isabel.
Okay.
Yeah.
I just wanted to ask a few questions.
Okay.
I think some of it we discussed a bit yesterday about measuring the carbon. Why do you have to send samples to Kenya?
Yes, because we don't have the machine for the analysis.
And
they use infrared, so that is why we sent it there.
Okay, you can't do the Walkley Black method?
No, this is gas.
Yes, it's for the...
So for this one, for carbon, we use CN analyzer.
Okay.
What I presented is emissions.
Oh, it's the emissions. Okay, no, then that's fine.
Yeah.
That's all right. Thank you.
You're welcome.
We have an equipment to measure emissions on the field, but that one is field built. This one, when you incubate and you have dispensed into the container, then we export to Kenya for the analysis using the infrared analyzer. But we are working to acquire our own infrared soon. Thank you very much.
Any other question, contribution, comment, addition?
Okay. Yeah, Joshua, thank you very much for the clarity in your presentation. In fact, I like your presentation so much. Thanks so much. And the land use systems, you made mention, you said there were 12, but for one or two reasons, you chose 10 or so.
Yeah.
But I think you presented only 5. Or the 10
yes,
also, was it a selection?
Okay, let me clarify this.
Yeah.
We have 5, but being replicated twice. So if
they are laying a new cable because you have.
Okay. So we have the DAI, which is the further production, and then we have the ecology, then we have the carbon, then we have the reference, and then the optimize. There are 5. Okay, it's showing. Let me show it. Yes. Okay. So the DAI, you see the first one, you see one DAI, meaning is the first rep. Then the last line down there, you see 12 DAI, which is the second rep.
Oh, okay. Okay.
So the only one I couldn't sample is the number 3, which is the vegetable. So when we come the second rep is the number 10.
Okay.
Yes.
All right. I think well noted. Okay. Then you made mention of the fact that you had higher yield with the oat. The oat gave you in terms of yield.
Yes.
I think the oat and then the grass also.
Yes. So when you are comparing the grains, the field, there is wheat, barley, oats, faba bean, and grass. So if you exempt the grass, and then you are using the grains, the one we human being we can consume, yes, it's the oat that gives the highest. But when you are comparing the grasses, is it the DAI, which is the further production system, the third year one, which provide or which produce higher than or significantly higher than the other system grasses.
Oh, okay. Yeah. But do you have reasons for that? Any reason?
Yes, I can't. Per literature, you see, the longer it stays on the field, it's able to produce more because of the, let me put it this way, I think Dr. Kelly wants to say something. Okay. Yeah. So the third year, to the best of my knowledge and the few literature I've reviewed, is because at that time it is well established. the establishment is there. So it provides a chance, and also, do you say one, two?
No, go on.
Okay, so that is the best I can say.
Okay.
Yeah.
No problem. I think I'll take it that way.
Because of time, I would encourage that we take it during coffee break. We'll still subject him to more input, questions, and so. Thank you very much.
Thank you.
But Joshua, just quick one. When this one, I think you should contact David and Abad on some of this. It is about the fibrous root system. Yesterday, I was discussing this with you that if you have a system where roots, the fibrous roots of the grasses are building up quickly, probably annually. In Ghana, the problem we are having is because of the fires. Within a year, you are getting about 10 fires, so these grasses are unable to really grow. Yeah. So if you have this thing building up and then decomposing into the layers, to be frank, when you came to Hungary, when you look into the profile, you could see that some of the grasses grow even up to 120 centimeters, some were extending even further. So if you have some of these things building up continuously, they will be adding carbon to.
Yeah. Thank you for the additions.
Thank you very much, Joshua. Can we give him a hand of applause? Interesting presentation. I think he's preparing for the Viva. With this, I'm seeing very good signs. Do not suffer too much.
So we invite all of you for the Viva next year.
Yes. We'll invite Manuel Beidu to come and give us a presentation on soil improvement in African Darkhead, enhanced carbon sequestration, soil nutrient status, and maize yields in Ghana. Shall we give it up for Emmanuel Sidmans, the program? Let's try this.
Okay. Good morning.
Good morning. So as you have 15
good morning to you all.
Can we show his slides?
It's there. So, control room, if you can show the presentation.
Can you show the presentation?
It's loaded, but control room.
Yeah, I've opened it here, so unless the control room.
It's there now.
Okay, so I'm presenting on the topic, soil improvement in African Darkhead, enhanced carbon sequestration, soil nutrient status, and maize yields in Ghana. It is widely known that human activities over the years has resulted in climate change as a result of emitting more greenhouse gases into the atmosphere. But in recent times, we have also found out that certain human activities in ancient times has also resulted in improving soil nutrient status and also building the carbon stock. And in this era of climate change, we see a very interesting phenomenon which has widely been reported in the Amazonian terra preta. So in the 90s, we see a group of Western scientists who were anthropologists, James Fahid and Melisha Leach, embarking on this darkhead concept in Africa, where they discovered the African darkhead. Interestingly, we had a group of scientists also in Africa, in Ghana, being led by our Honorable Professor Vincent Loga, doing a similar concept on the forest island phenomenon. So we see this as a great opportunity in building soil carbon stock and also improving our soil health status. So how are these darkhead created? Through human activities such as deposition of waste materials around, especially homestead in those ancient times, broken pots being burnt and other artifacts. And as a result of these activities, they resulted in a highly stable carbon-rich material in this darkhead. So we see in our exploration concept, we found how the nature of the darkhead were in Asante Kwa and also New Banfa here in Ashanti region. as you can see. Certain human artifacts that were also identified. You see broken pots, and you see pot materials. Oh, it's unfortunate you can't see, but I see it here, so we'll still move on. The questions we were trying to ask ourselves is what has been done as far as the dark earth are concerned. We see that in terms of the emissions of greenhouse gases, there is no data to support how much these dark earth are able to emit greenhouse gases into the atmosphere. Though there has been a report on in terms of the soil nutrients and high stable pyrogenic carbons in this dark earth. And also there is no information as far as how this dark earth can help in sequestering carbon in terms of the persistent nature of the carbon that are in the soil. And also there is a problem of converting this dark earth to other land uses because there is not much information as far as the stakeholders and also the communities are also concerned. So we see it as a great challenge that we need to bring on board to the Ghanaian community. So we justify this work by the concept that maybe the carbon-rich nature of this dark earth could be a great innovation for building climate-smart agriculture in sub-Saharan Africa. And also as a great initiative for the 4 per mil initiative, which is on the recarbonizing concepts. And building also soil organic matter would help to improve crop yield and also livelihoods. The main objective of this work was to help us understand the soil biogeochemistry. And I remember back then in US, Alan was still discussing this with me, that there is a need for us to extend this project as far as the biogeochemistry of the African dark earth is concerned, because not much work has been done. But specifically, we want to quantify the emissions as far as the dark earth and the non-dark earth ecosystems are concerned, and quantify also the pools in terms of the mineral associated and then also the POC as well. And then lastly, to estimate the yield potentials of these dark earth and non-dark earth ecosystems. So the study was located in 6 areas, from the southern to the northern belt of Ghana, where we have one, the southern belt, we have one at New Bonfa here in Ashanti, and then the middle belt, we have Sogloboy and Asantikwa, which we'll be visiting there, God willing, tomorrow. They are in the Kintapo North district of Ghana. And then you have the northern zone where we have...
Show the map. We can't see the map.
Oh, it's here, but sorry. I think the problem should come from... Okay, it's here. So this is the map of the study area. So in each of these locations, what we did was to select the dark earth, and then we look at a natural vegetation, which could be either a savanna or a fallow land, and then compare in terms of the ecosystem services that these are providing in terms of carbon sequestration and also soil fertility management. So within these ecosystems, we divided a 40 by 40 square meter within them, and then sampled within the 20 by 20 subplots. And then this is the sampling design. within the 20 by 20, we sampled from 5 different locations and bulk and take a composite sample per each of the subplots at a depth of 0 to 10 up to the 1 meter depth. So this is me and my colleagues and some farmers doing the harvesting on the field at Sogloboy. And then the greenhouse gas measurements on the field too as well, as you can see in this. Sorry. So in the
pause for some 2 minutes. So as you pause for 2 minutes, let me make some housekeeping announcement. Our field trip to Kintampo is tomorrow. It's strictly by registration. So we have our international partners all registered, our local scientists here all registered. We have 3 more slots, which is open. Yes. So if you want to be part of those 3 to join, we give you the opportunity to write your name on the decks. So we'll leave at 5 a.m. at the Impact Building. And then because it is more of October is very sunny in Ghana, I was actually telling Abad that you would have enjoyed your stay more if you were to come here in August. Very cold weather. So we advise if you have a sun hat, you can just take it along your field boots. And then we have dug a profile pit, which you have a look at the African Dark of Horizons. And then my senior colleague, Thomas, will lead that on the field. And then we have invited farmers to also join the trip. And then we can interact with the farmers and then visit very 2 notable areas on the walkway and also on the waterfall. And then we'll make a return journey back to Kumasi. So Caleb has asked me to lead the team, so we are all leading together. So he's still part of the team, and then we'll be, working as a team. Thank you very much for your attention. So, Emmanuel.
I think your intervention is timely. Everything is set now. Okay. So you show pictures of you on the field.
Go to where we have
this one.
The harvesting. Yeah. Okay. So this was how the harvesting was done on the field. And then we estimated the yield from both the dark earth and then the non-dark earth cropland ecosystem. So in terms of the greenhouse gas measurement, we did in situ, and I inserted these chambers on the field, and 3 chambers per each of the subplots, and then sampled using the syringe, and then placed them inside the gas vials, which were transported to Nairobi for analysis. Next slide. Okay, so laboratory analysis were done on these, on the samples, and also greenhouse gas measurement was also done as indicated in the slide at International Livestock Research Center, Nairobi, Kenya. Next slide, please. Data analysis, so results and discussion. So in terms of the soil carbon stock across the 6 locations, we find the dark earths showing higher stock in the 0 to 10 centimeter depths. At Navrongo, giving us the highest carbon stock of around 70 megagram per hectare. And this is very good for us in terms of carbon sequestration and climate change mitigation. And we see the African dark earths at New Bonfa also giving us around 50 megagram carbon per hectare. So the highlighted with bars with asterisks all indicates the African dark earths sites. And when you compare to the other ecosystems, you can see that dark earth sequesters more carbon than the other land uses. The same trend as in the 0 to 20, as you can see, and then up to a deeper depth of 1 meter. You see more stock in there in terms of the African dark earths. And then, interestingly, you see fallow land also exhibiting quite number of increase in terms of the carbon stock at deeper depths as well. So the pools, we have the mineral associated also giving us a very high concentrations in terms of the African dark earth. Oh, please, next slide. Next slide, up to slide 21. 21. Okay. So the mineral associated pool for the surface soil, which is 0 to 10, you can see the dark earth also having higher pools, higher concentrations in the mineral pools as well. Next slide, please. So in general, you can see that as the stock increases, you are also having more of the mineral pool, also having more of the carbon concentrations in there. And they stabilizes as you move down the soil profile. Probably because in most of our soils, you see clay also increasing down the profile as well. And in the last slides, you will see how clay also play a major role in terms of driving the soil carbon stock and also correlating with the stock as well. So POC also follows a similar. Please move to slide 24. A similar trend. But in terms of New Bonfa, we see high POC because of the management practices that is being observed there, which includes the application of both the organic and inorganic manure, and it is under cocoa plantation. So I think that in the short term, it is very good for the crop, which is cocoa. So the same trend in the 10 to 20 centimeter depth up to the 30 centimeter depth, where you have more of the POC pool in the at New Bonfa, which is under cocoa plantation. Slide 27, please. So this is on the stabilization. Slide 27, okay. So as my brother Joshua already said, I wouldn't waste my time here because you always have more of the carbon being stabilized in the mineral pool, and you have it contributing more to the TOC, the box soil carbon, than the POC in most cases, except where you have the fallow land and the cropland having more of the POC in there. So the similar trend where we see the S indicates savanna. So savanna also exhibits a little bit of, and I think it's interesting to my good friend Hamani, exhibit a little stabilization in terms of the mineral pool in those areas as well than the cropland, which is losing more of the mineral pool in terms of the stabilization. And you see, as I said earlier on, they stabilize more as you increase soil depth. So in terms of the grain yield, you see African dark earth giving you higher yield than the non-African dark earth ecosystems, because probably you have soil improvements in those soils than the non-African dark earths. So the correlation, we try to show how key soil variables correlate with soil carbon stock. And moisture, total nitrogen.
Can you go to that slide? You can see.
Slide. Yes. 31. Okay, is this one. So we see various soil predictors that correlate well with soil carbon stock. And the interest here was on the moisture, BC total nitrogen, then the CEC. So all those that were significantly correlating with soil carbon stock, I used them to get the regression model using the linear function. And CEC, clay, and then sand and total nitrogen were the key soil predictors that were driving the stock across the site and also across the land use system. But interestingly, you see CEC and then total nitrogen contributing about 30%. So finally, African dark earth savanna and fallow land stabilizes more carbon and nitrogen in their mineral-associated pool than their non-African dark earth cropland ecosystem. And this should prompt us as food security people who want to ensure food security, agricultural land that are losing more of the carbon in terms of the mineral pool. We need to be conscious of this and how we can help to resolve this crisis. As I said earlier on, CEC and total nitrogen as were key predictors driving about 30%, which means that the remaining 70% could be land use change, or it could be climate or vegetation types. So I am not saying that soil is driving all of them, because my political people are here, and fire could also be a major driver as well. So 30% coming from soil predictors, CEC and total nitrogen. And then lastly, I would say that the improvement of African dark earth soils increases crop yield than non-African dark earth, making African dark earth as a nature-based solution to improve food security and enhance climate change in sub-Saharan Africa, where they occur. I think that for climate aspects, our concept is not only on producing food, ecosystem monitoring, and also enhancements for environmental sustainability should be our priority as well. Thank you.
Thank you very much, Emmanuel. Shall we do that for us?
I'll acknowledge my funders and then my supervisors for this work. They've been a very great contribution to this work.
Who are your supervisors?
I showed it in the earlier slide. So go to the first slide. So Professor Loga, Dr. Caleb, and Dr. Yeboah, and then also Alan Planty. Okay. They have been the drivers behind this success you see. today. And then the EJP. Chavi, thank you.
Thank you very much. Any feedback, response, questions, inputs for him? Clarification? Or because he has thanked us, we are okay?
Okay, let me...
Sure. Yeah.
Joshua, Emmanuel, you were indicating that probably the soil contributes about 30% when you are looking at variability. Are you talking in terms of PCA? That's the first part. But I also want to say this. Do you think that because when we are dealing in soil modeling or PCA analysis, we have so many soil parameters down there, and each of them are going to contribute some certain number of, in terms of percentage, variance. Yeah. So at the end of the day, if you have about 30 parameters, 15 or something, and I'm going to look for how much they contribute to ecosystem variability, then probably I'm not going to get, in terms of maybe looking for maybe 3 key parameters, which will give me something above maybe 50%. It becomes much more or less. What do you think about this kind of, when it comes, is it that probably right from the beginning when you use a mixed effect or some modeling, or maybe you do some small general linear correlation, then probably you knock some out because of coloniality, you knock some out before you do, so that you will be able to see the real contribution of soil to some of these ecosystem.
Okay. So in terms of, let's say, the emissions, I did something like that, but I'll focus on what I've presented today, which is on the carbon stock. We are looking at what are the drivers, and per the results I have is only on soil parameters. So in terms of the soil parameters, pH and total nitrogen, available phosphorus, C, all of them. In the model, what I did was to bring all of them together, then run a linear mixed effect model. So in the model, the ecosystems and then the reps were fixed as random effects, including the sites or the locations. And then all the other parameters, which is the predictors, the soil predictors as a fixed effect. So what I did was that then I tell those variables that are not highly correlating or that are highly correlated, that is the multi-collinearity with a very high variance inflection factor. I did one. So get them out. If they are highly correlated with each other, I set the VIF at 10, which is the least. And some of them, for the regression model, the least I was having was 2, which is good for me. So if you have a very high variance inflection factor, which means that you are highly correlated, and as a result, you affect the outcome of the model. So I did a stepwise and backwards regression to eliminate those factors out. So at the end, you have the most significant predictors. Then you use it to get the model at the end. So at the end, I also want to know how much each of the variables that are significant, how much each of them is contributing. That is where the partial regression also comes in. So I did the partial regression, and you are able to see at the end, and in the regression results that I presented, you see how much each of them is contributing in terms of the total final significant predictors that I had.
You use the Limit function, right?
Yeah.
So with the Limit function, sometimes the problem we are having is they have a default of about 80%, meaning that if 2 parameters are correlating, and correlation is above 80%, it will also knock it out.
Yeah.
But for example, some of the predictor analysis we are doing, sometimes we are much more interested in parameters which are giving us more than 90%.
Okay.
So what do you think in the future you could do so that probably, the machine is having a default of the 80% mark.
Yeah.
So what do you think you could do? I'm just playing the devil's advocate here.
All right. I think for every predictor is good. We don't eliminate them completely, because I see it as being biased. If you take a particular variable out from the initial state, but as you progresses, you will see that those variables that are not important, the machine by default will be getting them based on the model that you are using.
Okay, in my thing you can see Caleb after, but my concern is that are you only focusing on soil chemical properties in terms of your modeling or relationships? I didn't see you mention any physical properties.
Yes, Prof. I think in terms of the texture, I brought in the salt.
What about the soil specific area? I think you did that parameter.
Yes, I did it, but Prof, I'll come and see you on that later, because there was a problem that was why, and I didn't want to contradict myself. I wasn't sure because it was giving me a negative correlation in terms of the stock that I was observing.
Did you use stock or you use carbon itself?
No,
carbon as a concentration.
I use the stock.
Okay, we can discuss after the meeting.
Okay.
Chimando, our next two presentations, we'll take two from our online audience. Are they there? And our technical people, whilst we are taking the online. Caleb.
Coffee break.
Break is at 11. Oh, okay. I don't know if they are evening.
They are in, but let's say that.
Let's take them because of the online. Yeah. So our online audience, technical people, can you switch us to online? We are taking the next two presentations from our online audience. Yeah, online. About the future, I see. It's Hamisi, can you hear me?
Yeah, I can hear you. Hello? Hello?
Hamisi is not there, then
I can hear you.
Maimela Maxwell from South Africa, Hungary.
I can hear you.
Is that Hamisi?
Yes, this is Hamisi.
Was that Hamisi?
This is Hamisi, I can hear you.
Okay. Then kindly share your screen. And hello, Hamisi.
Yeah, I've already shared my screen. Are you able to see it? Hello?
Hello, can you hear me?
Yeah, I can hear you. Are
Yes.
you able to see my screen?
And not yet. So give us 1 second and then you can start.
Okay.
Hello, Hamisi. You haven't shared your screen, so share your screen.
Oh. Let me share it again.
Yes, I think we can see now. Great. So you have 15 minutes. You can start now. Thank you. Can you hear us, Hamisi?
Yeah, I can hear you. Good morning, everyone.
Yes, you can start.
All right. Good morning, everyone from Hungary.
Good morning.
My name is Hamisi Samamkuzi. I'm a doctoral candidate at the Hungarian University of Agriculture and Life Sciences. And today I'll be sharing insights from my research work on the Kayakauma forest along the Kenyan coast, basically linking forest regeneration dynamics with soil, carbon, and climate resilience. So on this presentation today, it aligns well with the EJP soil workshop theme on the regenerative practices and soil carbon storage in Africa. So Kayakauma is located in Kilifi County. It is on the southern eastern part of Kenya. It is on the southern eastern part of Kenya, as you see on the pin. I hope the map is visible. It is a sacred forest governed by the Mijikenda community and recognized by UNESCO for its cultural and ecological importance. Like many coastal forests, it faces pressure from deforestation, encroachment, and of course, climate variability, which affects the regeneration and soil carbon stability of this forest. So understanding how this forest regenerate across the disturbance gradient, it will help us design sustainable and regenerative forest management practices. As you will see the forest, this is the structure. I mean, this is a sketch of the forest. Study context, the forest, in this research, I divided the forest into 3 ecological zones. That is, we have the edge zones that are highly disturbed and exposed to human activities. And second, we have the intermediate zones that experience moderate disturbances. And lastly, we have the core zones that are least disturbed with closed canopies and minimal light. So by comparing these zones, we can see how natural regeneration responds to environmental gradients and what this means for soil, of course, carbon accumulation. So basically, this presentation is guided by, generally, I was looking at to determine the regeneration patterns and the implication for soil carbon storage and forest resilience in Kenya. And specifically, we measured regeneration densities across zones. And secondly, we compared seedlings and sapling distribution by regeneration class. And third, we interpreted these results into relations to carbon storage and sustainable management. The study forms part of my PhD broader goal, that is to integrate ecological and cultural knowledge in forest restoration. In this study, we used a nested subplot design with 40 subplots labeled A1 to J4. That is, so I was having A1, A2, A3, and A4. They're moving to the B, B1, B2, B3, B4, up to J4, distributed across 3 zones. So the regeneration was classified into 4 broader classes based on height and diameter. And for class 1 and class 2, this was the category for the seedlings and younger regenerants. For class, the 3rd class and the 4th class, these were the saplings. So basically, from class 1 to class 4, the measurement, the range was between 0 to 150 centimeters height. So main counts were computed for each class and zones to understand the gradient of regeneration intensity. So first results, the total regeneration by zone. The results show a clear hump-shaped regeneration pattern. That is, the edge had approximately 312, which to me is a little bit moderate, followed by the intermediates that had a total count of, this is a tree count. This is a total regenerant count, and we have a total of 315 individual trees. Then the core zone, we had a total number of 270, which is the lowest regeneration compared when you compare the three zones. So basically, this suggests that regeneration is most vigorous and moderate disturbance supporting intermediate disturbance hypothesis, where too much or too little disturbance limits recovery. So our second results, the regeneration by class from the first to the last class. So when we break it down by regeneration class, class 1 and class 2, that is the seedlings, dominate across all zones, particularly in the intermediate zone, as you will see. Sorry, these are just averages, so you will see the decimal places. Of course, we can round them off to the next, to the nearest integer, the nearest whole number, I mean. And so in class 3 and class 4, the saplings, we see there are fewer, but they are consistent across zones, suggesting that fewer seedlings are progressing to, older stages. This shows active recruitment, but limited transition to mature stage, possibly due to light competition in the core and stress at the edge zone. So then another bit of the results. So looking at the seedlings vis-a-vis the saplings gradient, and basically from the summary here, like you will see that seedlings peak in the intermediate zone, and we have over 220 regenerants. That is 128 plus 95, so it's roughly 220 individuals on average. And on this other hand, in the intermediate, this for the saplings remains fairly stable across all zones. It's close to 90, 60 plus 31 in all the in all the edge. In all the zones. Like you see, the number, the tally is close to 90. So basically, it's fairly stable across all zones for the saplings. The pattern demonstrates that recruitment is strongest where disturbance and shade are balanced, while overly open or dense conditions suppresses regeneration. An interpretation of the results, basically the findings show that the regeneration dynamics follow ecological logic. That is, intermediate zones have the ideal light and moisture balance for seedlings establishment, while the edge zones faces exposure stressed, and of course some human impacts like the grazing, fires, etc. And on the other, lastly, on the core zones, we have limited light and thick litter, slowing recruitment. So these spatial patterns highlights the importance of canopy heterogeneity for continuous forest renewal, and of course, soil carbon inputs from litter and root biomass. Ecological and policy implication of this work. This results emphasizes the need for balanced management, that is maintaining moderate disturbance for optimal regeneration and soil carbon buildup. Second, integrate sacred forest governance into national climate and soil strategies. And lastly, we can use buffer zones as regeneration corridors to sustain both biodiversity and cultural values. So the Kaya forests offer a model for community-based regenerative management that enhances ecosystem services and local livelihoods. So to sum up, Kauma has demonstrated that moderate disturbance enhances regeneration. The intermediate zones are a key to sustaining both regeneration and soil carbon storage. Combining traditional ecological knowledge with modern restoration practices broader restoration and carbon management initiatives in the Eastern Africa and in Africa as a whole. So up to this far, I would like to acknowledge support from the flagship research group program 2024 Hungarian University of Agriculture and Life Sciences, the Mikenda Communities for Cultural Stewardship, my supervisors, Norbert Borish and Marta Fuchs, and of course, my, I would say, I will always run to Caleb for guidance. He used, back then when he used to be admitted, he used to be my first supervisor. So we still work closely with him. I do consult with him a lot. And yeah, that's it from me. I thank you for your attention. I look forward to your questions and discussion on integrating cultural heritage forests into regenerative land management strategies. Thank you so much.
Thank you very much, Hamisi. So any question, comment, feedback on the presentation? Okay. Hamisi, looks like everybody understands or there are no clarifying questions.
Well, I will be worried.
Yeah, so thank you for your presentation. We are grateful for
well, thank you so much. You're most welcome.
Thank you. We move on to the last presentation for the morning session before the coffee break. We are sorted in. So online. Maxwell, are you online? So the Modiba, Maimela Maxwell. Can you connect? I see you are online, so maybe you can unmute.
Am I audible now?
Yes, you are audible. You can share your screen.
Okay. Can you see it?
Not yet.
Not yet. Is it visible now?
Yes, we can see it now. So you have 15 minutes to take us through this presentation, then will be followed by some 5 minutes of questions. So take it away.
Okay. Thank you. Greetings to everyone, facilitators, and all the community of the European Joint Program. My name is Maxwell. Sorry. My name is Maxwell Udiba. I'm a PhD candidate also at the Hungarian University of Agriculture and Life Sciences. So I'm here today to present the findings of my study, which is titled Impact of Tillage on Soil Physical Properties, Crop Yield, and Quality. So these are the insights from a long-term experiment that started in 2002 in Hungary. And I am being supervised by Prof. Marta Birgash, the late Prof. Marta Birgash, who sadly passed away, and Dr. Barbara Shimon. So this study begins with soil health. We all know that soil health plays a significant role in agriculture productivity and sustainability. And among various soil management practices, tillage has long been employed for various agricultural purposes. However, the intensive mechanical application can also alter the soil structure along with its ecosystem services. Our study was done on a Shenazem soil, which we all know that it's among the most fertile soil in agriculture. However, even this kind of soil is not immune to physical degradation under continuous conventional tillage. So my aim was to understand how different tillage systems affect soil properties over time. And this is essential for optimizing sustainable crop production by employing tillage systems that are site-specific. So the study has a few objectives, which I will not go into the research questions. I'll just outline the objectives, which is specific objective number 1, which is investigate the impact of tillage practices on soil physical properties, which we focused more on soil moisture content and soil penetration resistance. And number 2, we wanted to assess the tillage effect on crop yield. Number 3, explore the combined effects of weather, tillage, soil physical properties, and crop yield. On number 4, we wanted to identify tillage methods for soil health and water dynamics. Determine tillage methods enhancing overall crop productivity. So because of time, all the results were not put in this presentation. However, the study was, as I mentioned before, it was done in Hungary. So the experimental area is called Joseph Major Experimental and Training Farm of the Hungarian University of Agriculture and Life Sciences University. So the soil type is an endocalcic genozem, which has a clay loam, which is a clay loam, and it has a pH that was tested in water of 6.1 and soil organic of 21 grams per kilogram. The study is a randomized block design for replications, 6 tillage treatments, which were disking. The depth of disking was 12 to 14 centimeters. On the table here, I don't know if you can see it, I've listed the implements that we used and also the depth of soil disturbance and the tillage methods. So on column 1, you can see the tillage methods that we employed, which is disking, shallow cultivation, no tillage, deep cultivation, plowing, and loosening, which I would like to clarify that shallow cultivation and deep cultivation, we used the same implement. The only difference was the working depth. These are the three types of crops that were planted. So it was a crop rotation of winter oat, sunflower, and winter barley. And the data collection methods, we used an HCalCom penetrologer to measure the soil penetration resistance. And we used a soil moisture meter, which is a PTA gauge, a Sarvach type, which is it was produced in Hungary to measure the soil moisture content. The crop root weight was measured, which is the fresh root weight, and the grain yield was measured when we were harvesting with a combine harvester, and also we measured other yield quality parameters which are not included in this presentation. So as you can see on the pictures here, denoted by the letter B, that's
So, can you hear us? Hello, Maxwell.
A generalized linear mixed models. And after analyzing the data where we found significant differences, we separated the means with a Tukey HSD post-drop test at 5% level, 5% alpha. And the results are as follows. So the moisture, the moisture content was measured from 0 to 50 centimeter depth. On my right, you can see I've shown a width graph, the soil moisture content. This is just to display how this the moisture content fed by treatment, by tillage treatment in the soil horizon. But the statistical results of winter wheat are shown on the table on the second column after the treatment. So we can see that the soil moisture content was lowest in loosening treatment, and the other treatments, they did not really differ much. In terms of sunflower, also from 0 to 50 centimeters, we also can see that the moisture content, however, was different compared to the winter wheat, whereby the treatment that had the lowest moisture content was plowing. And the graph also shows how the moisture content differ in the soil profile. The spatial moisture content dynamics of my study, which this one is winter wheat soil moisture content at various depths. So after measuring the data, we also aggregated the data into 10 centimeter depth. So to separate, to see the differences, if there are differences by soil depth of 10 centimeter. So in the 0 to 10 centimeter, we can see that we did not see any difference. And in the 10 to 20 centimeters, we can see that loosening had the lowest moisture content compared to no-till, which had the highest, and it was also significantly equal to disking. In the 20 to 30 centimeter, we can also see again that loosening had the lowest soil moisture content, and disking and no-till had the highest. We can also see that we recorded the lowest soil moisture content again at deep cultivation, and at 30 to 40 centimeters, we can see again that the soil moisture content was lower compared to other treatments at loosening, especially compared to plowing. The 40 and 50 centimeter also again that loosening had the lowest moisture content. And in terms of the sunflower crop, which was planted in the season of 2020-2021, at 10 centimeters, we can see that the soil moisture content was lowest in plowing, and in the 10 to 20 centimeters, the soil moisture content was also lowest at plowing, and at the 20 to 30 centimeters, again we can see that loosening and also plowing had the lowest moisture content as compared to no-till, and we at 30 to 40 centimeters, loosening and plowing again, they had the lowest soil moisture content compared to no-till. And at the 40 to 50 centimeters, there was a slight increase in loosening, which differed to significantly differed with plowing, and plowing had the lowest soil moisture content. In terms of temporal differences, we also measured the soil moisture content and the soil penetration resistance at 30 days intervals, which is monthly, on a monthly basis. And we can see that loosening also on the 14th of March had the lowest soil moisture content. And on the 11th of April, again, we can see that actually there was no significant difference. All the tillage treatments were the same in terms of statistical analysis. And on the 3rd of June, the loosening treatment had the lowest soil moisture content. On the 28th of June, again, we can see that there was no significant difference in terms of differences among tillage treatment. On the 15th of August, also, there was no significant difference recorded. So the temporal soil moisture content dynamics of sunflower showed us the following results. We can see that the lowest was found in plowing compared to all the other treatments, and on the 9th of September and on 23rd of October 2020, also we can see that loosening and all the treatments did not differ on this particular month. And again, on November 17th, the moisture was the same in all treatments. And
okay.
And on the 24th, also we did not see any differences. In terms of the penetration resistance from the 0 to the 50 centimeters, we can see that loosening decreased the penetration resistance in under wheat and under sunflower. Also, again, we can see that both plowing and loosening decreased the penetration resistance compared to the other treatment.
Okay. Maxwell, about 2 minutes to round up. If there are questions, so at least you take it because of time.
Okay. Yeah. Okay. Because of time, I'll jump the results of the the of the different depths, and I'll just go to show also I'll just highlight on the temporal of SPR dynamics of sunflower that was measured on the 2020-2021. So we can see that here on these graphs, from graph A to graph E, we can see that majority of the penetration resistant was high in disking and no-till, which shows that the disking and the no-till increased the penetration resistance compared to the other treatments. And also the monthly data also indicates the same thing that we observed with the different depths. And in terms of crop yield, we can see that several tillage treatments did equal in terms of grain yield. As you can see, the shallow cultivation, the deep cultivation, and the loosening for wheat, which is graph A, and B. We can also see that loosening it's still quite high compared to the other treatments. And this graph B also represent the crop yield of sunflower. So just to conclude, we can see that disking and no-tillage increased soil penetration resistance. Moreover, this affected the yield. And we can also say that these two tillage treatments, which is loosening and ploughing, proved more beneficial in eradicating soil compaction. And in terms of seasonal changes, with the trends with variations driven by climate and soil conditions, with notable increases during drier periods. So we also noticed that the penetration resistance is affected by the soil moisture, which increases during periods whereby there's less moisture and higher temperatures. And in all, we can recommend that a periodic deep tillage will be suitable in areas whereby they have drought and less rainfall, but they are practicing shallow cultivation tillage methods like your shallow cultivation and no-till. And regarding the soil moisture content, we can see that in terms of the temporal ones, it's the April and June, whereas in loosening improved moisture in April and June. This is important. Why? Because as I mentioned, that the crop has different soil moisture demands as it grows. So different stages requires different moisture content. So to know which tillage will be beneficial for that period, we notice that loosening it's good for crops that need moisture in April and in June.
Okay. Thank you very much, Maxwell. I'm sure when the questions come up, some of the clarifications can be given. Okay. But it's an insightful presentation. And as you, take us through the references, I'll give it back to the audience.
These are not the references, it's just some of the publications that included the work.
Hello, Maxwell.
Hello.
Yeah,
good afternoon. Yes, I can hear you.
Thank you for your very nice presentation. With respect to your deep tillage,
yes.
What frequency would you recommend for deep tilling? Should that be every year, or you would want to recommend tillage rotation? Because I know in some literature, you can plow in a season, the next season you skip plowing so that you don't have to use a lot of traffic on your soil and compact in that regard. What frequency would you recommend for deep tilling as per your results? Thank you very much.
Okay. In terms of literature, when you mention deep tillage, when they say deep tillage, they are using plowing as an implement for their deep tillage, and some they are using subsoilers.
You check your mic or technical if you can increase the volume a little bit. We can't hear Maxwell.
Okay, can you hear me now?
We can.
Can you hear?
Yes, we can.
Okay. Okay, so as I was saying that deep tillage in different literature, some use plowing and some use subsoilers. So to answer your question, if you're using a subsoiler, it is not economically advisable to loosen your soil with a subsoiler every season, because it is expensive to apply that. But in terms of conservation, it is very good that if you are on no-till or shallow cultivation, to also to check your soil. If there is a higher resistance in your soil, then you can apply periodic subsoiling. In terms of plowing with a plow, if you want to deep till or deep plow every season, that is not advisable because if you are plowing to the same depth, let's say 45 centimeters, below that depth, you will have a plow pen, and also you will be turning most of your soil. So from 45 going up, you will be turning your soil, and you will lose a lot of soil organic matter. So I would say periodic subsoiling, it is the way to go.
Thank you for the response, Maxwell. I see Caleb and then Johanna. Okay, Johanna can go.
So thank you for your presentation. You talked a lot about the moisture content. As I understood, you measured the moisture content once, right?
How do you mean once?
Once per field, or do you have different measurements at different times?
No, there was three. In one treatment, we measured 3 different spots randomly, and then we averaged.
Okay, and did you also measure the water holding capacity or the soil texture?
No.
Okay, because I think on your moisture content, that would be interesting to measure.
Okay, thank you for.
You're welcome.
Hello. Okay, Johanna, I think.
Yeah, hello. Yes, a quick one. I wanted to find out, looking at the data which you took your measuring, the intervals are not equal. Is there any reasoning behind, as in differences in dates, as in the interval? And then also, when your first table, you showed the mean, that's the moisture content for the mean, comparing the winter wheat against sunflower. I was of the view, if you could have projected the mean for the winter wheat and then the sunflower across, so that your point could cut across. Yeah, that's all.
If I'm not sure if I got your question correct. When in terms of the 2 crops, are you saying that I should compare the 2 crops? Because we are looking at these are 2 different crops. and in 2 different seasons. So hence we are not comparing the crops per se, but we're comparing the moisture content by season under different crops.
Hello. Yes, you compare the moisture for the different crops. So what I'm saying is that instead of you compare the wheat to sunflower, why not wheat, sunflower, and the oat?
Why not the sunflower and the oat?
Would it be possible if you could project where you had the crops you use against the season?
Which slide number was that? Okay, I'll start from the beginning and you will let me know where to stop.
Yes.
Where?
Where you had, there's no slide number, so where there is the winter wheat and against the year. The experimental design you use and the replication.
Hello.
But it's okay.
Probably I will help Max a little.
Yeah.
In this particular work we did, we were not much more interested in comparing the different crops because we were doing them at different times and different seasons.
It's not about comparison of the crops. I'm looking at, you see, the focus is on the moisture content.
Yeah.
So if the moisture content are presenting results where you are only comparing the moisture content over against the wheat and this, it's a suggestion, it's a view. It would have been more easier to understand for we who were not involved in the study.
Oh, okay. The whole idea is you are going to deal with editors, and then if I'm using 2 different seasons, and I'm going to say one is better than the other,
yeah,
that's a whole problem. So you don't want to get into that kind of zone.
Okay, then the other issue.
That's what I said.
The question about the interval, the interval in measuring dates.
Yes, I believe you are referring to the 28th and the 3rd of June and 28th of June, right? This one. Am I correct?
Yes.
Yeah, the thing is the experimental design, if we have rainfall and it's too wet to go in, the owner of the experimental site did not allow anyone or anything except animals that can enter. So when the moisture is too high, we cannot measure. Hence, you can see that this was measured in on the 3rd and 28th of.
Okay.
Yeah.
Thank you very much, Maxwell. I'm sure you can discuss it during coffee break. He's here. So let's take about 10 minutes. Let's give it out a round of applause for our online audience too. It's been an insightful presentation so far, and the interactions has been very good. So we'll take about 10 minutes coffee break and resume 15 minutes. Okay, 15 minutes coffee break and resume at 11:55. 11:55. Lunch is at 12:30, so let's do 15 minutes and come back. So to our online audience, give us about 15 minutes and then we'll come back. Thank you very much.
Okay.
Okay. Yeah, it's sharing. Yeah, great. We can see your screen now. But what you have is figure 2, so check if you are sharing the right screen. Okay. Page 8.
Yes.
Okay. Nice
Okay.
one. Great.
Kimberly, maybe you can close the tools window on the left side. Sorry, my left on the screen. That one where all tools, AI assistance, generative summary, because we are seeing that. Thank you.
Great. Good afternoon once again. Thanks for coming back from the coffee break. Next, we have Kimberly, who is making a presentation online. And she'll present on, I'll just give it to Kimberly, chemical fractionalization of organic matter in African dark soil. So, Kimberly, 15 minutes. And after the 15 minutes, 5 minutes for questions.
I don't
Over
know
to
if
you. Yes, we can see your screen.
Yes. Okay.
Great.
Great. So let me come here.
Okay. So by popular
That's
request,
good for
they
you?
said you can do the first few minutes for a quick introduction of who you are, and then the presentations can continue. Thank you.
Okay. So hello, everybody. I'm Kimberly Fulador. I am a master's student in soil science at the Federal University of Rio Grande do Sul, in Porto Alegre, Brazil. And I will present to you the chemical fractionation of organic matter in African dark earth soils. Oh, I had to. So African dark earth are carbon-rich anthropogenic soils that they are built by the past people and their management, principally by fire. And so we have the build of soil organic matter that have soil organic matter that has more stable molecular structures and like humic substances as humic acids and principally humin. So the objective of this work was extract the soil organic matter, including humins and humic acids, from African dark earth soil samples collected in Ghana, following the methodology developed and previously validated by members of this proposal as Hayes and co-workers 2008 and Song and co-workers 2008-2011. So the soils were collected in 3 sites. The first of them, Ashantikwa, where is grown cashew and plantain. And the soil there is a luvic phasem soil. The second site is Sogliboy, where is grown maize, principally. And the soil there is a pratic anthracite. And then we have the third site, that is New Bonfa, where is grown cocoa, plantain, and cassava. And this site I visited, and is a site that is not managed for a long time. I don't know exactly, but since decades. And there we have a lychee soil. So here we can see the map of Ghana, and these soils, Ashantikwa and Sogliboy, are localized in the Bono East region, that is more central region in Ghana, and New Bonfa is localized in the Ashanti region. We can say also that in the south of Ghana, the further south of Ghana, we have more tropical climate, and the further north we can find some more dry areas. So all of them, all of the sites were collected, the soil samples were collected in 3 depths, 0 to 10, 10 to 20, and 20 to 30 centimeters in pseudo-replicates where the samples from the 4 sites were collected at each depth were combined. And then we originated of 9 samples because we have 3 depths and 3 sites. So we have 9 samples of air-dried fine earth soils. And here I want to show you in the table 1 the carbon content of the soils through depths. So we can see here that Ashanti-Kwa and the Soliboy have a very similar content of carbon. And New Bonfa has the higher carbon content here. And if we see the soil characteristics of them, we could think that maybe New Bonfa has the least favorable characteristics to sequestrate carbon, but maybe it's sequestrating more carbon because of the management, because we can I can remind you that, for example, in Soliboy, they grow their maize that is annual cropland, so maybe there they till the land and they turn over sometimes, and this management can be the carbon can be mineralized, so there's a loss of carbon by CO2. So here in the table 2, I want to show you that is the 0 to 10 layer. And we have here through the depths, through the sites, the humic acids and the humins that were extracted in each step of this. So the sodium hydroxide at pH 10.6, sodium hydroxide at pH 13, sodium hydroxide plus urea, and then the humin that's soluble in DMSO and the humin that's insoluble in DMSO. So we noticed that we can observe that New Bonfa has the higher yield in these steps, in these extractions, but we also see here that as the extraction goes forward, the extraction of more persistent, of more complex structures, the yield decreases. Precisely because maybe in the soil, there are more, in these soils, yes, there are more labile structures that are less complex structures than persistent ones when you compare them to each other. And however, when we compare humin fractions, here the humin soluble in DMSO and insoluble in DMSO, we observe that all of them show a greater amount of more complex humin, that is the humin insoluble in DMSO, compared to the less complex one, that is the humin that is soluble in DMSO. And here I want to show you between the layers. Yes, the yield. And just something that I want to say to you is that I had some problems in extracting the with sodium hydroxide 10.6 and 13. And just to compare, I unite them, but you please have in mind that it is separated. And I'm conducting another batch here in Brazil that I will be able to fix it. So for the other layers, the first layer, the 0 to 10, is okay. But, so we can see here that Ashanti-Kwa and New Bonfa, through depths, it increases, right, the yield. And because of the characteristics of the soils of these two sites, we can maybe, it can be maybe because of the clay elevation that we have there. And maybe these humic acids, they are associated with clay. I don't know, but I think I didn't. This one is first, okay? So to understand something about the humic acids, so here the extraction steps of chemical fractionation. So I extracted with sodium hydroxide at pH 10.6, at pH 13, and sodium hydroxide with urea. And these two first, the pH 10.6 and 13, we could collected the humic acids that they are more susceptible to decomposition, or maybe they are more labil, if you want to talk in these terms, or they are less complex. Yes, something like that. They are these. I'm talking about their structures. So here with urea, that urea helps to break the hydrogen bonds. So we could collect in this step with urea the humic acid that are less susceptible to decomposition, that are less, yes. So the humin here that are structure that is not, how can I say it? It's not soluble in aqueous media. So we had to use DMSO, the dimethyl sulfoxide, and we could collect it, the humin that is less. All humin is a complex structure. Yes, it's more recalcitrant, if you want to say in these terms. But we have a humin that is soluble in DMSO, that is a little bit less complex, and a humin that is insoluble in DMSO, that is a little bit more complex. Sorry about that. So maybe here you could understand better. So here, going on, we have the humin in DMSO that through depths, okay, it increases in the from 0 to 10 to 20 to 10 to 20. It increases, then it decreases for the last layer. And the humin insoluble, it still has to be performed the last the humin that were collected in the 10 to 20 and 20 to 30 layers. Here is another way to see this data. Then if you want, sometimes the other way is better, sometimes this way is better. Here we have the depth, and in each step of extraction. So the first one here, we have the sodium hydroxide with the 2 pHs, and then we see that it has the more yield, the higher yield. And then we have the urea and the humin. So some considerations that we have here is that New Bofa has the higher content, even though it's the soil that has the least favorable characteristics for this. And maybe it's because of this management. Even though persistent organic matter fractions, such as humin, they are present in the soil, of course, a larger proportion consists of structures that are more susceptible to decomposition. Therefore, proper management is essential to prevent carbon mineralization. Another thing is the first layer, the 0 to 10 centimeter samples have already been sent for NMR analysis that will be able to see the functional groups. And once the final batch currently being processed is complete here, that I'm conducting, the remaining depths will be analyzed as well. And the last is that I intend to present these novel findings of humin and humic acid fractions from African dark earth at the International Humic Substance Society meeting that will be held in Czech Republic in the next year. So I thank you, and I am here to answer some questions that you have.
Thank you very much, Kimberly. So questions online. We can start from our online audience. If you have any question, you can unmute and then, or contribution or comment, unmute and make the comment. Melissa, Lauren, if there are any. If not, we'll head back to the auditorium and take the general comments.
Congratulations, Kimberly, for your work. Thank you for presentation. Are you from Brazil? And it's very, very good to see a Brazilian student presenting. Who is your advisor in Brazil?
Yes, sorry, I could be, but Etelvino from Embrapa. And Deborah Pinheiro Dick is my is my professor, and Fabiane Vezani. This work is not from my thesis, my master's. But my master's, yes. But I went to Ghana this year, and I developed this work there.
Oh, great. Great. This is very interesting. I know all your advisors. They are very, very good. Thank you. No, I just need to know who are advising you. But thank you very much for your presentation.
Thank you.
Thank you, Melissa, for the comments. I see Dr. Yabua.
Yeah, Kimberly, thanks for presenting the work that you did in Ghana. And Melissa, this work was initiated through Etavino and Deborah as part of our ongoing research initiatives. So we decided to host Melissa, Kimberly over here, and she spent quality time to generate the data, and we are very, very happy with what she was able to do. Now, she has presented this already to us, and we still need a lot more understanding of some of the follow-up work to be done. You would be looking at the NMRs from the samples that you sent to Brazil. But I realize you are focusing on the 0 to 10 centimeters. If I looked at your last slide, you will be focusing on the 0 to 10 centimeters. So what happens with the other depth? Are we likely to see the MRR functionalities from those ones as well? So that's number one. And then when we move to slide 8, I would like to probably understand the various pH ranges that you use, 10.6 and 13, as part of the extraction process. What was the basis for these pH ranges? Thank you very much.
I think I'm lost here because I can't.
Can you go back to your slide? If not, I think, but the important thing is the basis for the pH.
Slide number 8.
So, okay. So, as I understood, what happened is that, for example, I think in the pH 10.6, we have some structures that are more labeled, right? But if it is not in this pH, if it's like 10.3, for example, it didn't extract it. So in the other layers, in 10 to 20 and 20 to 30, it didn't extract it well. So in some of the batches, it extracted, but in some of them, no. So what I did here to present to you, we have an idea of it. I united them, but I am conducting another batch with 3 gram soil to pick this problem so we can have this it separated, right? So it's just a way. It's the way to see different forms of what? Different forms of humic acids. You know, humic acids that is a little bit more soluble, label, and the other ones that are a little bit less label. Sorry. So I don't know if I could answer you, but I think that's the case.
All right. But I still want to understand for the 0 to 10 and then the 10 to, in terms of the signatures for the NMR, what you are going to do now in Brazil.
Okay. So with NMR, I already sent the first layer with the insoluble humin to them to analyze it. And I think that as we have a human, insoluble human that has a higher content, that's a higher yield than the, for example, human that is soluble in thimerosal, we will find a lot of aromatic structures that they were probably built by fire, right? By the way that ancestors, peoples managed the soils. So these first layer were already sent to NMR analysis, but now I have to, how can I say it? But to finish this other batch that I'm conducting, to have this pH 10.6 and pH 13 separated, so I can send to them to analyze this correctly. And the human insoluble, I already made the HF, the fluoridic acid treatment to clean it. And it's just, I have just to weigh this, and it's done.
All right, thank you. Please, just one more and then I'll be done. You have done the chemical fractionation, and our students in Ghana have also looked at the physical fractionation. How easy would we be able to compare the results from the chemical fractionation and then the physical fractionation? And who will take the next steps to be able to, if you want to do this, comparing the chemical fractionation of this African dark Fs and then the chemical fractionation, which we have never done on our African dark Fs in Ghana?
I think there are two ways to look at the surrogate matter. I never did the physical fractionation, but I think that both analyses could be used to look at this with at a view of soil organic matter of African dark earth soil samples, and to, how can I say it, but to
yeah, I think the supervisors, we can discuss this together with Deborah.
To have a better characteristic, yes, to have a better characteristic of these African dark earth soil samples.
Thank you, Kimberly. I'll take one more reaction or question. If there aren't, we'll move to Lorraine. Any other question? I think we've done well. Lorraine, are you online? Let's, Kimberly is a I forgot. In fact, the interactions has been very, very exciting. Kimberly, thank you for the great work. Lorraine, are you online? Yes, Lorraine, thank you. Try and share your screen. Let's try your mic. Can you say hello?
Hello, hi, I'm Lorraine.
Great, we can hear you and see you. Kindly share your screen.
Okay. Can you see my screen?
Yes, we can see it.
Okay. Hi, good afternoon, everyone. My name is Lorraine Martha Mita, a recent graduate from the Hungarian University of Agriculture and Life Sciences in Hungary. My research thesis was on the effectiveness of agroforestry.
Lauren, just one moment. Can you put it in presentation mode? We are seeing your presenter screen. So display settings. Yes. And click on it and swap. Swap presenter view and slideshow. Yes. Click on it and then swap presenter view and slideshow. That one. Select that one.
It's not working.
Okay. Duplicate.
Okay, let me stop sharing and try again.
Yeah, try again and let's see. Yeah. Okay, this is good. So 15 minutes, Lorraine. Please take it away. Thank you.
Okay. Hi, everyone. My name is Lorraine Martha Minta, a recent graduate from the Hungarian University of Agriculture and Life Sciences. I had my thesis on the effectiveness of agroforestry system in mitigating climate change through carbon sequestration in temperate regions, and the study was done in Salvash in Hungary. So, as we all know, African agriculture faces a dual challenge, that is feeding growing populations while also meeting climate commitments. The industry needs solutions that build soil health by also sequestering carbon. And this research was from Hungary's temperate agroforestry system and offers proven methods which is directly applicable to African tree crop systems. The study was tested in Salvash, which is a region with a continental climate, with hot, dry summers and also moderately cold winters. The study site featured alternative rows of trees and also crop alleys, with the tree rows designed to function as the windbreaks and organic matter contributors. My main research objectives were to assess the impacts of different agroforestry systems on some soil properties in Salvash, and also to quantify the effectiveness of different agroforestry systems in sequestering soil organic carbon across various depths. I used a randomized complete block design with 4 treatment zones relative to the tree rows. That is the tree row, the buffer south, which was on the sunny side, the darker shade, which was the buffer north, and the open alley crop, the middle. I had 3 sampling depths, 0 to 10 centimeters, 10 to 20 centimeters, and 20 to 30 centimeters. I analyzed the soil pH, electrical conductivity, the carbon content, the bulk density, and organic carbon content. And I used a two-way ANOVA in R to test the effects and depth, to test the effects of depth and treatment. So for the soil pH, from the figure here, you realize that the tree row had the lowest pH of about 7.6 at the surface, that's 0 to 10 centimeters, by increased at the deeper level, which is 20 to 30 centimeters. The middle and the buffer south had a consistent higher surface pH with a minimal change across depth. The buffer north slightly dipped as a mid depth, that's the 10 to 20 centimeters, but it followed up to an increase to 7.9 at 20 to 30 centimeters. So for my soil electrical conductivity, there was a decrease in the electrical conductivity at the 10 to 20 centimeters, followed by a substantial increase at the deeper level, that's the 20 to 30 centimeters. The middle layer had the highest electrical conductivity, while the tree row and the buffer south had a stable electrical conductivity profile across all levels. For my bulk density, the bulk density showed a slightly increasing level at the middle and the buffer south. The tree row displayed inconsistent mid-depth values, and the buffer north remained stable across depths. So the soil organic carbon concentration had the highest surface soil organic carbon at the tree row. But at the Papa South, it maintained a high soil organic carbon across all the depths. It had a moderate soil organic carbon at the middle zone, but recorded lowest at the buffer node. For my soil organic carbon stock, which was my main focus of my study, the tree will show the highest soil organic carbon at 0 to 10, but decline with depth. And the buffer south maintained a uniformly high soil organic carbon across all the depth. The middle had a moderate soil organic carbon throughout the profile, while the buffer node consistently recorded the lowest soil organic carbon values. So for my research, I assessed how a temperate alid cropping agroforestry system in Salvage was going to influence key soil properties and carbon dynamics by integrating trees with crops and enhancing soil organic carbon and improving soil structure. The soil properties such as the pH, electrical conductivity, and bulk density remained stable, indicating there was no negative soil alteration under the tree influence. Carbonate content supported carbon stabilization in the alkali soils, and soils closer to the tree store significantly higher carbon stocks compared to the open field areas. The south-facing buffer zone accumulated more carbon than the shaded crops, highlighting the role of microclimate effects. So since I'm having this presentation in Ghana, and though I did it in a temperate region, the relevance to Africa was that the lessons from temperate Hungary are transferable to Africa systems like cocoa, cashew, maize, agroforestry systems. By promoting tree crop integration, it can also boost store carbon fertility. It can reduce erosion and increase water retention, and also supports natural climate integration. To conclude, agroforestry enhances soil carbon sequestration and also soil health without compromising productivity. The key message here is the trees on farms are climate allies. The long-term monitoring and scaling across different agroecological zones will further strengthen evidence for carbon-based incentives. And I'd like to thank all the research. I would like to thank my university, the Hungarian University of Agriculture and Life Sciences, my supervisor, Dr. Mihai Yancho, and Dr. Caleb Okansi, and also the Department of Irrigation and Soil Science in Salvage.
Thank you very much, Lorraine. So if there are questions or feedback or comments online, we can start from there, and then we'll come here. Any comments?
Yes, I have. Good morning again. I have just some questions about how many years that had that experiment.
I did a year. It was for my master's thesis.
Just 1 year. Yeah. How many trees are there? I mean, I would like to know when that experiment was installed. Before your masters, installed or sown before your masters, or just for your masters?
So if I get your question well, how long has the experimental field been?
Yes, thank you very much. How long?
Okay, so I think probably 2 to 3 years, because there was already a PhD student having a research on that particular field. And I collected the soil samples and she was there 3 years prior to I came. So
okay, because yes, my question is because the tree seems it's adult, the trees. Mm-hmm. So I'm thinking because we need time to observe carbon sequestration and soil. Yeah. Here in Brazil, we have, including public policies, giving an incentive to install this kind of systems here. But we are expecting have more information about carbon physics, or more I don't know, more change in carbon stocks in the soil through the years. So because of that, it's very important to mention how many years since the trees were planted, or how long the experiment has, and the cultures. I didn't know which culture we have there. Just between the trees, what did you planted there? It's just grass or maize?
Yes.
Maize.
Was maize.
And before maize?
I have no idea.
This is important for your discussion. This is very important. Because of that, we can thinking which kind of differences we are gaining with the soil, including biodiversity, because of the previous culture. So this is important when you write your material, you're bringing this information. When the trees were planted, since when we have cultures, which culture we had before mine, and then I think that this is historical information. This is so we have sometimes we have more. We need more details previously than that the information that we have in that time in the present. Okay, just have this information. It's very important. Okay, thank you. Congratulations for your work.
Thank you so much.
Very much, Melissa, for the comment and then the feedback. It's very important. Yeah. Any more from online? Kimberly, you are still there. If you have a comment, you can make it. Or if you have a question for her, because you have already presented, so you can't ask you any more questions. So that's fine. I see a hand up here. Lauren. Right, let me take him down. Come here. Your mic
works. Thank you. I just want to highlight, since there are questions surrounding some historical information that you need to get to support your auxiliary data. Have you, perchance, found out if satellite services could be of support to provide you with such historical data, maybe 2 to 3 years or possibly 5 years, the type of crop in that zone? Develop an airshed for the satellite to pick that particular zone for the past years, and maybe even in seasons, then you can now get that historical data to support the work you are doing. Thank you.
Okay, thank you very much.
Yeah. Lauren, thanks for sharing this insightful information with us. Kindly go back to your concluding slide and have a few things for us to discuss. The concluding slide, I think the point number 1, if you can share.
This one?
Yes. The very first conclusion that agroforestry enhances soil carbon sequestration and soil health without compromising productivity. I would like to know the productivity indices that you measured in this work. And then also, when you talked about the soil health, what parameters I was listening through your presentation, and I want to get the soil health indices that you used to be able to come up to this conclusion. Thank you.
To answer your question based on what I just said about, it was basically about the trees on farms, our climate allies, meaning the integration of trees on the field with our crops, helped us measure carbon to the soil and also improving the soil structure. So the integration of the trees and crops helped improve the soil structure of the land. And this also aligns with the global importance of agroforestry. I don't know if I've answered your question about the soil health.
I see. Okay, Caleb.
By building soil carbon, it builds soil carbon and also improving the soil structure by maintaining moisture.
Okay. So, Lauren, I think what we will need to do is you have to contact Mihai, for some of the data we're talking about. But the whole thing about Lauren's work is they've been using technology, that is drone imagery, to take a lot of agronomic data on an experiment they've been doing for over a decade. So when I got to Hungary, the idea was, can we try to assess what they did in the past was they take the soil profile and then, do routine analysis for each of the plots, and that is all. So she was the first to do a real soil analysis that is in relation to carbon sequestration on that particular field. So all the historical data we are asking from her, there's none. She was the first.
Thanks, Kali, for the clarification. Any more questions? Thank you, Lauren, for the presentation.
Thank you.
Please give her a round of applause, please. She's done well. Okay. So we take the last presentation from Mr. Lemuel Wabeng. Just a moment, let me see. Okay. Let me see if view. That's full screen. This one, yeah. Yep. So wait, and then. So Lemuel's presentation will build up on Dr. Aidan's presentation yesterday. We've seen a lot of soil, so now we are going to animals. So, and gases, one more, and then. We need more animals. Sorry. Okay, so Lemuel, 15 minutes, and then.
All right, so my name is Lemo Wabinsian, just like Prof said, and I'm with DEPA Lab. And in DEPA Lab, we have worked on a lot of innovations, and some of them which can help us control and then fix these carbon emission problems that we are facing today. So one of the devices that we have worked on is a device we call HeadWatch. And HeadWatch is a smart livestock monitoring device that is going to help with the low carbon future we are all seeking. Now, we know cattle alone contribute to a large amount of methane and greenhouse gases from their enteric, from their activities, their digestion. When they die, their defecation, they release carbon materials into the environment like methane gas. Now, how do we continue to raise these livestock? Because they produce food for us. But then if we leave them just like that, they are going to continue to impact our environment, which we are trying to fix. How do we manage these livestock so that they reduce the environmental impact they have on us? And that is where HeadWatch comes in. What is HeadWatch? HeadWatch is a simple device think of HeadWatch as a smart watch for cattle, right? So here we see that the device has been strapped at the neck of the cattle there, and that is taking some data. So HeadWatch is equipped with technologies like GPS, temperature sensor, motion and activity sensor, and then heart rate monitors. That is going to take data from the cattle. Now, the GPS is going to be helping us to track where the animals go to graze. Here, we can use this to control the overgrazing. Also, temperature, motion and activity monitoring sensor, and then the heart rate monitors can help us detect diseases early in these cattle so that they do not die on us. And when they die, their body decay and then they release methane gas into the ecosystem. So this system is helping us to manage our cattle ahead very well. So, like I said, how do cattle contribute to carbon emission? Methane emission from cattle enteric fermentation. Like when they eat, their digestion, they release methane gas into the environment. And then emissions linked to inefficient grazing and land use also. So when we leave our cattle to just graze anyhow, they destroy our green plantation. And this plantation helps to remove some of the carbon release carbon that have been released into the atmosphere. So if we just leave our cattle like that to graze anyhow, overgrazing, and they destroy this plantation, we don't get to remove most of the carbon that are released into the environment. So this will help. This will lead to leaving a lot of carbon footprint in the atmosphere. And also, if, like I said, cattle are not managed very well, their health are not monitored, their activities are not monitored, they just die. And when they die, their remains release carbon into the ecosystem. Now, how is HeadWatch going to help us? I've mentioned this earlier, that early health detection for reduced weight. So the animals that have HeadWatch devices, when there's something wrong with their health, maybe their heartbeat slows down, their temperature rises, the owner of the cattle will know that, oh, my animal is not feeling well. And then the device will send those signals with AI-powered tools, will send those information to the farmer so that the farmer will know that this cattle is having a problem, and then they will attend to the cattle so they can save the cattle in time, that it doesn't die and becomes a waste. And then also optimize grazing and land management. So we have heard of geofencing. So we can create a point, area where this one is specifically for grazing for cattle. We don't leave them like that to destroy people's farms. We leave them at one place where we have planted grass for them to graze on. We control the grazing of cattle. And when we do that, we are going to help remove some of the carbon footprint in the atmosphere. Also, if a farmer is able to sit at the comfort of his home to monitor the animals, he doesn't have to drive to the farm to check on the animals all the time. He has everything on a screen, monitoring them in real time. He doesn't have to drive from his house to the farm using fuel, which also release carbon footprint into the environment. So HeadWatch also help here. Also, preventing stray cattle and vegetation damage. So in building HeadWatch, we had a lot of engagement with our farmers and stakeholders. And some of them mentioned that sometimes their cattle just enter into other people's farm and they destroy their vegetation. And that is a problem that HeadWatch is also going to help us solve. Also, when we're able to collect these data, we are not going to just leave them like that. We will make climate data-driven, smarter decisions based on the data that we are going to collect. So we know how data has become expensive these days and how data is helping with the machine learning and the AI world. So collecting these data will also help us a lot. Also, this data is also going to help the farmers to track their carbon removal so that they get some carbon credit, and then they also benefit from that provisions as well. So when it comes to the social and economic impact, Headwatch makes huge contribution to national and global climate targets, as we have explained here today. And then the farmers also benefit by reducing animal loss and increasing productivity. Imagine not losing your animal to disease, they not dying. You find out when they are pregnant early so that you take care of them. It is going to increase productivity, and you are going to, the farmer is going to make money out of that. And it also helps to introduce our local farmers to this new emerging technology like IoT and then AI. So we know some of our farmers, they will not use ChatGPT for anything. They don't have use for them. So if the AI is going to help them solve problems that are related to their work, then it is going to be beneficial to them. Thereby, we will not be leaving our farmers behind. Now, what are the current progress of Headwatch? So Headwatch is in production with Dipper Lab. So you can see me there in the Dipper Lab assembling the first prototype that we are looking forward to collaborate with the Animal Science Department on field testing. So we also went to meet some of the farmers at Afram Plains, where we spoke with them. We got their feedback on whether Headwatch is going to help them solve their problems, and we got positive feedback, and then we proceeded to building Headwatch. Also, the Dipper Lab software team are also constantly working on the software to build AI-powered software, which is going to help merge the hardware with the software so that the farmers can easily visualize the data and track their animals easily. Now, in the future, we are looking to deploy the Headwatch, but then for now, we want to collaborate with the Animal Science Department and then do some controlled field testing on campus. After that, we produce more prototypes, and then we take it to the farms. And then we go and then do more tests and collect more data on that. And then as we collect more data, we generate more AI-powered tools so that we can make predictions and make the device better. Then also, as we test the device on harsh environment, looking at where the cattle graze and all that, we are looking to improve the design of the Headwatch device so that it can withstand the harsh conditions with less, with less, I mean, you understand what I'm trying to say. Yeah. And we're also looking forward to collaborate with researchers across the globe so that they come help us build this idea and then bring it to fruition. Yes. So now we have seen that the Headwatch device is going to help us minimize livestock-related emissions. But then we can't 100% take that out. The animals will eat, they will defecate. What are we going to do to their remains? Now, that is where this device comes in. We call this device smart biogas technology. And what does it do? This device here is going to be attached to already working biogas plant. It is also a monitoring device, just like the Headwatch. Now, when this is attached to the biogas plant, it is equipped with temperature sensor, pressure sensor, humidity sensor, and methane gas sensor. Now, what are we trying to do with this device? We are coming to track the quality, the concentration, and then the pressure of methane gas produced. And then we will match it to its temperature so that we'll be able to look at the optimal temperature, the optimal humidity at which the biogas plant produce a lot of methane gas for maybe power generation or for cooking. So with the help of the smart biogas technology, our biogas plant will be able to work at its optimal condition, thereby producing a lot of biogas for farmers to use. So this smart biogas device is going to help reduce carbon emission as well. So the methane gas that would have escaped will be converted to energy, and it will be used in our households or in some schools, and then some industry too who have adapted biogas system. Then also using biogas will replace the use of fossil fuels, burning of firewood, charcoal, which release carbon footprint into the ecosystem or the atmosphere. With the help of biogas, if you're able to utilize the biogas plant very well, we'll be able to eradicate all these, and then we'll convert waste to energy. Also, the device, like I said, monitoring the biogas plant is going to help improve the biogas plant itself to work at its optimum condition. And then also with the help of the gas sensor and then the pressure sensor, we'll be able to prevent leakages. So when some biogas plants produce the gases and then in the pipes that they go through to maybe the kitchen or the biogas power generator, maybe there is a leak somewhere. With the help of the pressure sensor, we're able to sense that, and then it will be sending live data, just like HeadWatch. It will be sending live data to the user, and then the user will be able to make quick decisions on that as well. So with the biogas plant device, we've collaborated with DAS Biogas at New Japan. We've done our first field test, and with the help of our friends, 3Es for Africa, who provided us with the funding, we are also perfecting the design so that we will launch it officially as a product which will be used in the biogas sector. So in the future, we seek to scale across multiple biogas plants. We are not going to limit ourselves to just the DAS Biogas producer in Jabi. We are going to other regions in the country to test the device on multiple biogas systems. Then also, with the data that we are going to collect, we are going to use advanced analytics and AI optimizations to help make data-driven decisions so that we'll be able to predict the amount of biogas we can maybe produce in a month based on the conditions of the biogas plant system. Then also, we want to enhance the hardware for rural conditions. Like I said, internet connectivity and all that play a role in IoT systems, and we want to use the best communication protocol in some of these devices, so we are experimenting with some wireless protocols that will help best in these IoT devices as well. And then carbon emission tracking and verification. So we are integrating we seek to integrate carbon accounting systems so farmers and organizations can quantify the emission reduction, unlocking access to carbon credit and sustainability financing as well. And then we also look forward to partner with clean energy stakeholders like the government agency, NGO, and renewable energy companies to speed up the adoption of this system in our communities. So, in conclusion, we have seen that though animals produce food for us, they also contribute to destroying our environment. But then, with the right tools, we can manage these animals very well to minimize the emission and the impact they have on our environment. And HeadWatch and smart biogas technology is going to help us do so. So HeadWatch is going to help us manage our animals very well. And then SBT, smart biogas technology, is going to help us convert their waste to energy, which is going to benefit us all. So these are some innovations that at the DIPA Lab we are working on. We are open to collaborate with friends outside, and if you want to, we are open to funding too. Our doors are always open, and we seek to welcome you to help us bring our innovations to life. Thank you very much.
Thank you, Lebon. So, as he said, this is a collaboration we started with the animal science guys, Net, Yao, and then halfway, because of development, we paused, but we will continue.
Thank you. That was a good one.
I'll use it.
Animal science. You said you have been there. Who did you speak to?
For me, I don't remember the name, but my boss here knows the people we collaborated with.
Oh, okay, okay. That's good. Oh, okay, Pimpon. Oh, okay. All right, that's good. I am interested in how you are going to prevent the animals from going above certain radius or maybe distance that. How are you going to do that to prevent them from straying to other places?
Yes, so with the geofencing, fencing, first we demarcate a geographical area. with the help of the GPS trackers. When they go beyond that, so we are, the head watch is still in prototyping state, and we are looking to add some electrodes. So you see, there are some visual devices that when they are attached to your skin, you can initiate some small shocks to your skin. Very good. So we want to incorporate that system in head watch. So when the animal crosses the boundary, we give it a little shock so that it turns back. So that is how we are going to.
Okay, that's good. So in terms of the data measurement, you mentioned temperature. You mentioned what again?
Heart rate.
Heart rate.
Then we have motion sensor to monitor their activities.
Okay. So are you considering measuring methane directly?
Methane from the cattle?
From the animals. Because when they breathe or belch, okay, the gases they release is not only carbon dioxide. Depending on the enteric fermentation, there can be some methane gas coming out. Do you intend to capture that? Because we have some devices that already are able to capture methane directly when you put it under in the neck of the animals.
Okay, so for now, the headwatch device is modular. Modular in the sense that it is open for upgrade. So we will add those sensors to them. So for now, we are focusing on the health parameters and then the GPS tracking. Yes.
Okay. I think that's good. Thank you.
Yeah. We'll see you too. Animal science.
Yeah.
Yes, any other question? I see. Okay.
I have the mic now.
Yeah, you've seized the mic.
I'm a very, I'm always enthusiastic about technology. Okay. So I'm very happy that you guys are putting out this. I think it's going to be helpful. However, one of my biggest problems with proponent of technology is often how the technology simplifies the reality of life. So in this case, if you are a herder, you are more interested in finding forage for your livestock. You are going to Joe fence me when I have seen the grass on the other side. That looking delicious. You are saying I shouldn't go there. So I think that you have to look at that a little bit, because when I listen to you, I think what you get, some of the issues that will come out is what we discussed on Monday, the trade-offs, right? In terms of using technology to restrict animal movement, what is the implication for example, the herder or the farmer who is interested in fattening the cows and selling them during Ramadan or any period? But on a more technical level, what I would like to know is what pilot data do you already have? I mean, have you piloted this? And do you have some data on it that can speak to some of the potential challenges?
Yeah, so like I mentioned, our first prototype is still underway, and we are seeking to collaborate with the animal science team so that we do our first controlled field test on campus. And then after that, we will go to the farms.
Yeah.
Sure.
Cattle grazing is not only limited to the animal side of it, it also involves the vegetation aspect of it and the dynamics that happens. It's also, I think, interact with the soil that we have been talking about all morning. So I think you have to open it up a little bit more to the different categories of stakeholders that are likely to interact with this system.
Okay, so
thank you.
My boss is here. We'll look at that.
On the first point, as you rightly said, there are trade-offs. In fact, when we went to draw Bonso, we couldn't even meet the farmers and the herders separately. The farmers want restriction. The crop farmers, they want restriction. The herders also don't want restriction, but rather they don't even want to know the movement. They want to know, for instance, the female cattle, if they are pregnant. That's the only parameters they are interested in. So, but So technology, if you are developing, depends on the point of view of the users. So as he said, it's modular. So based on the application, it can be tailored to fit. If, for instance, there is regulation that requires control, then geofencing becomes important. But you know, our regulation is very loose. And as you said, in fact, within the team, there is Caleb, there is Enoch, and then there is animal science. We're thinking animal science were the major collaborators. But as you said, the crops, the forage, and even the methane component just came, I think, from Dr. Aidan's presentation. After his presentation yesterday, we're discussing if we can incorporate that. But I think there are a lot of avenues for us to collaborate, and then, yeah.
One last thing on this. How long does the device keep before going off?
In fact, so one has been done. It's called CSAM. It was piloted in the Alpine region of Germany, right? So with their calculation, they looked at something, if based on the range, what duration are you looking at? Is it from morning to about evening when they go, and then when they go back to the crowd, and then it can be taken and then it can be charged again. If not, there are various levels of complexities. If you are not doing onboard processing, that is processing on the board itself, then if all that you are measuring is data, then it can stay on for a very long time. If you are interested in processing on the field, then you can do maybe from about 6 a.m. to about 7 p.m. Then you charge it again. So it depends on the mode in which you want to use it and then how long it can last. I don't know if it answers your question.
It does. But so in the long run, we're looking at fixing every animal with this.
That was what we were thinking, but the cattle farmers, the headers, they only want to fix it on some selected ones, the leader, and then those by the side and those things. But ideally, if every animal, let's talk about in the ideal case, if every cattle gets it, then you can monitor every cattle's health and even
contribution so this is going to be a very what we call big data thing.
Yeah.
How are you going to process all this huge number of data that will be coming within a day?
Thank God. We have very huge server capabilities at the DEPA lab. And as Dr. Caleb said yesterday, we are, besides all the things that we are the knowledge hub for Carbon Extra. So we have compute infrastructure. We have very good compute systems to process this. Right?
All right, that's good. Thank you.
But of course, we still want more money to build our capacity. So the collaboration to build more clusters coming.
All right. I would like to talk to you guys maybe when everything is done here.
Lemoy.
Let me seize 4 minutes and then.
Yes, thank you very much. It's really good to see these developments, but I just wanted to make the comment that in New Zealand, we're using the halter system. It's getting much more popular now, and people are using it widely. And it's a system, exactly as you say, it keeps the cattle within particular areas with GPS locations. But the biggest cost that the farmers avoid is they do not need to put fences there. And fencing is a huge cost. And so so you don't need gates, you don't need fences, and that is a huge advantage to the farmers.
Thank you.
Management system are they using there? Is it like in the US where you have quite a huge area that you can keep your livestock? In our case, we actually don't have demarcations. So
we're not dealing at enormous scale, we're dealing at farm scale. But if the farmer decides that he wants to keep the cattle to a certain area to allow the grazing to occur to a certain depth and then move them to another area, then that's the way they're adopting it, and it's working perfectly well, yeah.
Thank you. One more comment, question, and then...
Lemoy, I wanted you to elaborate more on the geofencing, the advantages, for example, with the headers, the conflict usually between the headers and then the farmers. If you elaborate more on that.
Yes. So with our interaction with the farmers, we met on a trip at Afram Plains. The farmers are complaining that the herders bring their cattle to their farms, they graze and destroy their produce. And now when they go, you see, the herders are not the same people that own the cattle, the herd. The herders are just looking after them, so they just go and graze, and then they bring them back. So when there's any damage caused, it is the owner of the cattle that is supposed to settle the farmer. Now, if the farmer comes and complains to the owner of the cattle that this your animal came to my farm to destroy my produce, sometimes there is a conflict. The owner might say that they were not my animals, and it can cause a whole lot of problem, a lot of fights between the farmers and then the cattle owners. So with the help of the geofencing, we will have record, evidence, timestamp on where the animals went to graze. So the head watch will be able to help us. Well, they will not stop this point, it will not stop the farm, the head from destroying the produce, but then at least compensation to the farmers will do. Yes.
The discussions can continue during the lunch break. Thank you, Lemore. Can we give it up for him? So thank you very much. I'll take 1 minute to conclude. I think it's been insightful. And on behalf of the presenters, now I'll speak on their behalf, we'll thank the organizers for giving us the platform for the next gen and the current generation to showcase the work that we are doing, and for the feedback that you have provided, right? They say in Ghana, there is an adage that says, follow who knows road. That is, those who have done it before, your feedback into it will guide us to follow the correct path. So we are grateful for all those who stayed in and made valuable contributions. We thank you very much. On this note, I'll also thank my audience as the chair of this session for making it very exciting. Sometimes people make presentations and we sleep throughout. To the presenters and then the audience, you didn't make it boring. So we are grateful. At this time, we'll thank everybody and then we'll proceed to have lunch and reconvene. Caleb, after what time will we reconvene? 2 o'clock. It's 1:25, almost 1. We reconvene at 1 hour, so 1 hour lunch. If we can come back early, we'll close early so we can have the other session. So the online people too not. Thank you, everyone. Okay, bye for now. Thank you to our online presenters too. And then if Dr. Melissa is there and she wants to give a comment, it's fine. Okay, so then if you are online, your virtual lunch will be saved. We'll send you a picture of our lunch and then you can enjoy with it. Thank you very much. Bye for now.
Okay. Bye-bye. Thank you.
Lunch, it's always a challenge, but I know you'll be able to keep alive and let us have the rest of the presentations. So I will be talking about the Red Soil project in Ghana, which has good alignment with the ongoing EJP project, so that we learn from each other and see how we can make progress. So currently, I will present on behalf of the team, myself, Professor Andrew Sopoku from KNUST, then we have our FAO partners, Perata and Michaela from the NSLG, who is coordinating the project from Rome. The presentation will move along the introduction to the Red Soil project, the baseline results that we have, then some few conclusions, and then some announcement that will be relevant to you and to all of us. So now, what is the Red Soil? We are talking about recarbonizing of global soils. So that's the whole idea, recarbonizing of global soils. And if you looked at it carefully, it's an innovative initiative from FAO trying to work towards maintenance of soil carbon and increase of soil carbon. So if you are recarbonizing, you are looking at a situation where you can maintain what you have and then also try to increase it. But the moment it starts declining, then you are not recarbonizing. So this is the focus of the work that we are doing. And we work through trying to provide sustainable soil management. And we try to also work towards compensating farmers where necessary. And then it's also a technical support, mainly providing the sustainable soil management and then trying to really understand the soils that the farmers are working with and move towards providing a tailor-made management practices for the farmers. It's worked around maybe 4 pillars: a technical support, then incentives recognition, then we talked about sustainable soil management implementation, and then the recarbonization of the soil, and then the greenhouse gas mitigation. So it aligns very much with what we are working on in the EJP. So this is exactly what we work on. There are phases of the Rec Soil project. We have about 7 phases. The first one that we normally work towards is the identification phase. Then we work towards the preparation stages, and then the technical training for capacity building. And then we also look at the processes of the project areas, identification of the project areas, and then the baseline assessments. And then you looked at all the monitoring aspects and then the finalization of the project. So we started somewhere in 2023 on this project, and we've gone through all the phases. Currently, we are on the phase 6, where we are doing the monitoring. So we just finished the baseline data collection, and now about the monitoring. So I'll be able to highlight some of the baseline data for us. So this is where we are with our implementation. Now, I will talk about the baseline results. And why do you always need the baseline? If you are working on any project now, it's quite important to really take good steps to do the soil sampling and analysis. So the first one, we are looking at the sampling of the soils, which we always use a specialized mechanism to identify the sample locations. And then we do the analysis, the soil data chemical analysis, as well as some physical parameters. Then we try to put the results together to know what else we have to do. So that is where we focus on the management aspect. So knowing your soil, you are able to come closely with what will work based on the knowledge that we have in soil science to really address the problem. So once that one is done, we are able to improve on the productivity of the soil, which also leads into decreasing CO2 emission or putting carbon back to the soil. And if you monitor it very well, invariably, all that we are doing translates into the carbon money. So either from your crop, increases in your crop production, you improve your livelihood, or if you are able to put all the data together across all the farmers we are working with and demonstrate the carbon that we've sequestered, that also translates into money. So it's quite important to really follow the sequence of the documentation to really establish the baseline and all that comes along with it. Now, why is it important to have the baseline? The most important aspect of it is that if you do not have the baseline, that's the initial one, you wouldn't know what is happening. With time, So the first graph is just giving you the land use management that you have currently. What happens if you change the land use to land use system B? What happens? And then also, the second graph on the right is also highlighting after a period of time, from 0, maybe 4 years, 20 years to come, what will be the scenario? Are you going to have business as usual with your management strategies, or you can demonstrate that you are increasing your soil carbon stocks, or you are decreasing your soil carbon stocks? So it's quite important to establish the baseline and be able to monitor from time to time. And these are some of the issues that important for the baseline establishment, because from once that one is established, you know whether you are putting carbon back to the soil or carbon is being emitted. And we also know whether you are decreasing the nitrous oxide emission following direct application of urea or fertilizers. So it's quite important to establish the baseline for subsequent monitoring. Now, what is the baseline data from our work in Ghana? We sample from the Laura area in the Upper West region, and we were able to really get about 30 to 32 farms in those areas where we sampled, and we have 2 depths of sampling, 0 to 10, and then 10 to 30 centimeters. We also looked at the soil bulk density parameters, and then we have about 80 samples for bulk density, and then 40 samples for soil respiration. So the soil respiration comes in to help us assess the CO2 emissions as well as the methane and then the nitrous oxide emission. And then when it comes to the soil analysis, we talked about the soil carbon stocks, then the nitrogen, the phosphorus, CN ratio, texture, and then the basis, and then the exchangeable basis. So these are the parameters we've been monitoring from the sites. And we always use the NDVI we have the clusters using the NDVI, and then finally, relative elevation of the sites, just to be able to systematically know the sites that we are working on. Now, with the work that we have done, we realized we took the soil texture from all the 40 samples that we have analyzed, and by way of the soil textural classes using the USDA, we've identified that we have most of our soils being the loamy sand, which is about 64%. Then the next one that dominates is the sandy loam, and we have very little sandy soils in the area that we have, and then the sandy clay loam. So this is overall the textural classes that we are working with, giving us an overview of how we can manage our soils based on this information. Then we look at the soil carbon stocks at the 0 to 30 centimeters and texture, trying to really see the relationship that exists with the soil carbon stocks and then the various textural components that we have: sand, clay, and then silt, and then the soil plus clay fraction within the 0 to 30 centimeters. And when you look at the soil carbon stocks with respect to the clay content, you see a real, most of them are limited within the 0 to 5%. So that's where we have a lot of the clusters. But then you see differences when it comes to the sand fraction. So quite a lot are rather negative, correlated with soil carbon stocks. And that is what we are observing. So with the soil texture that we have taken, we are able to establish the relationship that exists between the soil carbon stocks as well as the various soil textural components. And this paper, I'm sure when you have time, you can really look at it, establishing the relationship between soil carbon stocks and then the clay and silt content, which is quite one of the good journals that will relate to understanding the soil carbon stocks and then the textural classes. So I'm sure maybe when you have time, you download this paper and it will really inform you as to how some of these things were done. But you realize that, yes, as the clay content increases, we have the soil carbon stocks also increasing. And that is one of the things that we need to get out of this. So again, when we look at the layer, the soil carbon stocks and then the texture, we realize that we have the soil carbon stocks is quite high with the loam soils at the 0 to 10 centimeters, but quite low with the sandy soil. So at least the data that we have collected, we can subject it along this line to see where we have more soil carbon stocks with respect to the various texture that we have. But then when we move also to the 10 to 30 centimeters, we realize that the loam soil have more soil carbon stocks compared to the other soil parameters. Again, when we also look at the upper layer, or the 0 to 30 centimeters, comparing the loamy sand and then the sandy loam, we find a lot more soil carbon stocks in the sandy loam fraction compared to the loamy textured sand. So these are some of the relationships that we can derive from the work that we have done. This paper also would be quite relevant, trying to have the soil carbon gaps and try to know the density that we have with the soil and then the soil organic carbon content, trying to establish the relationship within that. So this paper also will be quite relevant for the postgraduate students and then the master's students working on this, giving you a good understanding of the relationship between the density and then the soil organic carbon content. With our soil, we realize that when it comes to the soil texture and then the concentrations, we have the loamy sand and then the sandy soils. So we have the soil carbon concentration, and then we looked at the sandy soil and then the loamy soil. So this is similar to what I've presented earlier, but trying to make use of the data that we have, establishing the relationship between the various soil texture and the soil carbon stocks. Also, we looked at the 0 to 10 centimeters and then the 30 to 20 centimeters with regards to the soil carbon stocks, and we still have this same relationship being established. So at least within the soil carbon stocks and then the soil depth, we are able to establish the relationship between the soil organic carbon content or the soil carbon stocks and depth variation. There are other factors that we are still looking at which influence the soil organic carbon. So depth and then the aggregation is something that we are still exploring. But you realize that, yes, when we talked about soil carbon stocks and then the soil respiration, we have established this relationship, which we know that the bulk density under the soils with enhanced microbial activity is essential for really increasing soil organic carbon accumulation. And then the higher soil respiration is limited to greater soil organic carbon concentration and carbon stocks. So these are some of the outputs that are emerging from the data that we have. Again, when it comes to storing possible strong correlation there between the nitrogen levels and the soil organic carbon accumulation. We have a strong correlation from the data that we have. Then adequate nitrogen availability is essential for promoting soil organic carbon storage. And this is also coming from the data that we have. Soils with high soil organic matter really also tend to retain higher nitrogen levels. And then out of this graph, we can establish that we have An optimal carbon to nitrogen ratio of 10 to 12 appears to maximize soil organic carbon accumulation in the soils that we are working with. So these are some of the outputs from our baseline data, which now helps us to monitor what happens. So again, this thing had been already explained in the data. We now looked at the soil carbon and then the phosphorus. I was indicating to, I think, Jeron, that yes, we have very low soil phosphorus levels, and compared to what we saw earlier in the morning from their phosphorus levels, we see a contrasting issue. We have organic soil phosphorus levels quite low, around maybe 5 ppm, but from the sample that we have one extreme case which we are still trying to understand whether it's really deviant or we have made a mistake in our assessment, which is quite high. We don't get this level of phosphorus in our soils. But from the samples that we have, we are still trying to understand why we are having it. It's from one of the natural vegetation, but it's still completely high from what we have. So almost all sites seem to exhibit P concentrations which are less than 10 ppm of all the sample size, quite low. And then we have weak correlation between phosphorus and soil organic carbon. This is from our results. And then when you look at soil organic carbon and pH acidity, we don't see no strongly acidic or alkaline soils peak, and then we have the soil organic carbon near pH around 6. So that is where we are having our results. Around pH 6, this is where we have most of the soil organic carbon levels. And then there's no clear link between soil organic carbon and absolute exchangeable acidity. And then again, but we also see negative correlation with relative acidity from our graphs. And then again, we have low base saturation, which may also limit soil organic carbon accumulation from the results that we have. Now, again, we're looking at the 0 to 30 centimeters and then the pH and the acidity. We see the relationships that are quite negative correlation between the soil organic carbon stocks and then the exchangeable acidity as we have on the graph. We have also looked at the soil organic carbon and the bases. And these are the observations that we have, that high base contents and high soil organic carbon accumulation. And then we have strong correlation with calcium, which might also be an important consideration for sustainable soil management, which can really improve on the liming potential. Maybe for the want of time, I will skip this slide, but it shows the same components as before. And then when we look at calcium and acidity, we have liming with calcium may be needed to really increase the nutrient availability, but also pH to these soils would have to be looked at. So these are some of the observations that we are having. And liming with calcium may be needed to increase nutrient availability, but also pH in all these soils that we are working with. And then we, as part of the management of the soils that we have and the farmers that we are dealing with, we want to see whether there are major limitations for yields and soil organic carbon sequestration in the different farms that we have. Working on this, we looked at it in terms of available peat, and we have limitations for yields are quite low. So here we find out that in all the soils that we have, we pick each farmer and we try to look at the available phosphorus for each of the farmers that we are working with to establish whether we have low, very low, high, medium, or whatever content. Say that when you are providing sustainable management practices,
it is tailored to the farmer. We are not working with too large numbers of farmers, but the few farmers that we are working with, we provide them with real scientific basis based on the soil information that we have and guide them as to how they can manage the soil. So for available P across sites, we see very low P levels. So each farmer, we know the amount of P that you have and what are the options available to really improve on the available P. Then we discuss with them and come out with what we'll use to address or correct the problem. Now, when it comes to total nitrogen, again, based on what I've shown earlier, we have very low percent nitrogen on all the farmers' fields. And this is really, really a disturbing one, but it is not too different from what we see across most sites in Ghana. The nitrogen level is quite low, sometimes less than 2% in most of the soils, and we still need to work towards it. And then the pH, we realize that the farmers that we are working with, their soils are normally within the range of slightly acidic, around 6 to 6.5. That is where we have most of the pH of the farmers that we are working with. And then, with respect to total exchangeable basis, it's also very low. And this is typical of the soils that we have in our environment. So, but the whole idea is we are able to look at each farmer's site and the nutrients from the chemical to the physical and be able to establish what is it and how we can really address it. So we have the sodium alkalization, and which are also quite low, and we try to look at the implications. So trying to come out with some level of conclusions from the baseline data that we have collected. We can say that, yes, soil texture, most of our soils are very coarse, and the main limitation is low water retention. I mean, having a coarse soils, you definitely know that your water retention would definitely be much of a problem. And considering maybe drip irrigation and maintenance of soil cover is key. And these are the options that we discuss with the farmers, trying to see whether mulching, composting, residues, retention, and trees or grasses on their fields. But at least you have the basis to come out with the management practices that you want to propose with them. And then when you talk about carbon inputs, these soils depend strongly on carbon inputs to build soil organic carbon, from what we have shown. So we need to have the roots, the biomass, the compost, the manure, and then the residues. So in coming out with sustainable soil management practices, these are our first point of call, with the farmers deciding which of them they would like to go. With microbial activity, we've read that topsoil respiration and microbiology strongly influence organic carbon and soil health. And maintain residue cover for moisture and temperature balance will be an ideal thing to do in the soils that we have. And then we try to really improve on the mulching, the field application of biomass cuts, and then avoid biomass burning. And these are some of the things that we discuss with the farmers, that biomass burning is something that, if possible, we try to avoid. Then when it comes to the nutrient deficiencies, the NMP, we need to apply fertilizers and sufficient levels based on the soil information that we have. And then we combine the inorganic, the organic inputs to optimize CN ratio and better yields, and also work towards soil organic carbon improvement. With regards to the base cations, we've seen that the low calcium level that we have, and we consider liming or gypsum to increase the calcium levels in percentage terms. And then with possible soil deficiency, sulfur deficiency, we are looking at having sulfur-based fertilizers to be able to apply. And then when it comes to acidity, we looked at some moderately acidic soils that we have could also benefit from liming. But this is not coming from the blue. based on the information and the data that we have, we come out with some of these conclusions. So in concluding, we are indicating that, yes, understand the initial status of each production system and monitor changes over time. At least the baseline data is highly important to guide in the monitoring. So this is what we want to establish, and then provide data on starting conditions to define achievable goals. This is also quite important. So even if your work is within the PhD program, still establish the baseline for people to build on it subsequently. And then we also want to strengthen other tools and initiatives like the GSOC and then the other opportunities to do modeling with the data that we have. And then again, we try to ensure alignment with the MRV protocols, enhancing transparency and trust to enable project scaling. So these are some of the initial conclusions that we have based on our baseline data. And then detect constraints. What are the constraints of the farmers' soils that we have? Is it acidification, nutrient imbalances, salinization, or compaction? Then we come out with tailored soil management practices. So this is it. A solid first step to change our soils from soil organic carbon sources into soil organic carbon sinks. And this is how we want to progress. Establish the baseline and work towards improving the sources into sinks for climate change mitigation options. Now, there are a few things that are related to the work that you are all doing. In 2026, we have the 10th International Symposium on Soil Organic Matter, which will happen in Sao Paulo, Brazil. And my good friend Albert and the team are really the brain behind this. Please, the call is open now. Submit your abstract on time. Explore financial support to attend. And let's see, your abstract will go through and you highlight your work. And then again, the other one I want to highlight is the 23rd World Congress of Soil Science, which will also be from June 7 to 12, 2026 in Nanjing, China. All your work will find a way in these international programs. So thank you very much. These are the partners that I'm working with from the FAO, Michaela Brown, Gail Perata, myself, Andy, and all the other partners. So thank you very much.
Thank you very much, Dr. Yeboah, for the wonderful presentation. Please, we invite questions from the audience.
May I? Yes, Melissa.
Hello, Melissa, you can go first.
Yes? Okay, thank you very much. Congratulations for your presentation. It's very interesting to know how the farmers are, what is the real conditions of the farmers from Ghana. And I was thinking about it. I didn't see any data about integrated crop livestock systems in those areas, in fact, because if I know if to plant trees sometimes is hard, requires a huge investment, but maybe if some public policies exist, if these public policies exist, maybe could be a way to encourage these farmers to adopt this kind of techniques. So again, is it possible to encourage these farmers to use this kind of techniques to enhance soil organic matter through feces from the cattle, for example. And we could enhance phosphorus, we could enhance nitrogen from this management, and microbiodiversity too. Yes, I'm hearing. Thank you.
Right. Thank you very much. This is very, very good observation. And it's something that we discussed, I think, on the first day when we were thinking of the integration of the livestock into the farming systems. But it's one of the practices that they do, and also crop rotation. I never had a chance to show the sustainable soil management practices that the farmers themselves provided. But we normally work with them to understand the options available, and what they choose is what we now use to address their own problem. So yes, there are, in the traditional northern regions of Ghana, we still see the crop livestock integration, and it's a major farming practices that we have. So it's a point that is well noted. And it's something that has also been demonstrated that it can really improve the soil carbon stocks when such practices are implemented. Thank you very much.
Thank you. Just a question about the public in Ghana, encouraging this farmer to do it, or something like government money with kind of, I don't know the word for this, but some kind of borrow. In Portuguese, we use the word empréstimo. I don't know the word in English, but when the bank gives some money and the farmers need to give him back with low taxes. I forgot the word.
Whether there's incentive package for the farmers, and they can also have a low facility in terms of borrowing from the banks to improve on their practices. We have the NGOs that are doing very well in this area, but from the government point of view, I think that is a challenge. So, but the NGOs normally comes with the providing the logistics, either the inputs, they support them with the inputs, and when they sell their products, they are able to pay back along that line. But at the government level, it's the technical information that is given to them through the extension services. But when it comes to incentives, we see the NGOs doing a lot more than the government.
Johanna?
Yeah, thank you for your interesting presentation. I have one comment and I have one question. So first the comment, it's really great that you're able to work with so many farmers. I think in Germany they are often afraid that we want to change something or it will be costly for them. So I think in Germany it's hard to have so many farmers to work with. And for my question, did you publish these data somewhere, or are you going to publish it?
In fact, we have just finished our reporting to FAO, and we go through a series of approval from FAO before we are able to publish. So all that we have done is analyzing the data, and we are writing it out. By the end of the year, we will be able to get approval. Our report, as I sit here, I was working, my report is due 31st of, 31st of October to be submitted. So I'm working on the report. Once the report is submitted, approved, and we get a green light, our papers is also being put together. Approval given, we are able to share the data.
Okay, thank you.
You're welcome.
Prof, I have the microphone, so I guess I'll just elect myself. Prof, thanks a lot. That's a very interesting presentation. And it's also very refreshing to see that at least there's a support to build real data, because often the projects we get are quite small and doesn't allow for long-term data collection in areas such as this. So it's very interesting. I have one small concern. You see that our landscapes are predominantly from natural areas like protected areas. Then you have the fallows, and then you have the actual farms. And my concern is, as you presented, I don't know if that is the case. The data seems to come only from the farmlands. So I was just thinking about, it would be interesting to be able to compare these data sets at least under different land use system, and if there are plans for something like this.
Very good. Thank you very much for the question. And your observation is really, really correct. That the data is coming from croplands, from the farmers that we are working with, because we are focusing on the food crops and what are the options available for them to keep on their own fields. So it's quite an important one. But now we are moving a little further, trying to understand at the landscape level. So we have a tool being used by FAO, the Exact tool, which try to give the model aspects of what we are, and this one takes care of the landscape. So we sample from the landscape also and try to look at the different management systems and be able to provide the data as we have and still do the soil respiration, because without the soil respiration, we are not able to come out with the CO2, the methane, and then the nitrous oxide emission. So we are working on this now, and the model is giving us good outputs from the different management systems that we have. So yes, let our report be approved, and we are able to get our papers also approved to be published, and we are able to go share with you. Yeah.
Yes, I saw one of your recommendations is to encourage farmers to mulch using maybe residues. But are farmers willing to do that here in Ghana? Because in South Africa, we face a big challenge. Most farmers prefer to feed livestock using that residue. So to get them to use it as a mulch is a big challenge. So here, how are you navigating that issue?
Yeah, so from the conservation agricultural point of view, we discuss. We don't go with a prescribed management option for the farmers, but we discuss with them based on the results that we have. Looking at it, part of your materials would have to be fed to the animals for you to get the residues back, but you try to maintain at least 30% soil cover, which is also a challenge, but at least part of the residue, immediately after harvesting, they move into the dry spell, which normally nothing grows. And the animals feed on whatever is available. So it's still a challenge, but it's something that we still educate and encourage that at least part of your residue is left on the field. If you can maintain 30%, and then the rest of it is cut and carry, you confine the animals and you feed it to them. So it's not an easy task, but we don't fold our arms and say that it's a challenge, so you not start. So the education is ongoing. We are advocating for it. And some of the farmers now cut the materials, leave about 30%, and confine the animals and feed the animals with whatever is cut to feed them. So this is the practice. It's not an easy task, but we still need to move.
Thank you, Doctor, for the presentation. I just wanted to ask, I was just observing on some of the graphs that we are presenting, that most of them seem to be modeled using a straight line graph. Is that the best trend that you could use to demonstrate the relationship or the correlation between the parameters that you were measuring? Or maybe second order polynomials could be better? I was just looking at some, and I thought maybe a curved graph or an exponential graph would maybe describe the relationships much better.
Yeah. Thank you very much for the observation. And based on the outputs from the models and then the graphs, this is where we have. But we will still explore which one can really tell our story best. Because at this stage, we have moved from the single parameters that we have measured trying to understand. So we are not presenting them in a tabular form for you to see that this is the nutrient levels, but for each farmer, what is it? And trying to come out with the best way of presenting. But as you said, we will explore different ways. The one that tell our story better, we will follow with that. So I will meet you after coffee to discuss some of the options that are there for us to really explore. That would enhance our work and give some credibility so that when the paper finds its way to you for review, I know that I have already cleared myself. Thank you very much.
Can I have one
question?
more? Can I have one more? Yeah. Look, sorry, once again, you just mentioned something about dealing with individual farmers, and then I had this question before. So the question is, so we collected data from different farmers, and even though we assume that they are almost homogeneous in terms of how much resources they have to even input in their farming system, but we know that there's likely a lot of discrepancies among the farmers in terms of how much they do on the farm already in terms of input. Have you taken that into consideration so far with the analysis that you've done?
Yeah, so the resources available to each farmer is different. But at least each farmer knows his soil, his or her soil, knowing clearly that if this is my situation, and we all discuss that if you find yourself at this level, these are the options available based on the available resources within your community. So we will not come out and tell you that, yes, add poultry manure or cattle manure. What is available in your community? What are the options available? So now a lot of them are doing their own composting. And some are adding the biochar compost. If you go to the LoRa environment, it's one of the areas where biochar addition to soils is really moving very fast. We started, and now almost every farmer within those communities is producing his own biochar and adding to his soil management practices. So I understand they are not the same in terms of resources and then the endowments that they have. But knowing what you have to start from your soil, not from somebody's field, your own soil, and work it out to improve. Yeah.
Thank you very much, Dr. Ebo. I have a question for you, but we are late. So when you sit, I'll come. We now have Dr. Amani too.
Right.
Imagine going after Dr. Ebo. Good afternoon, everyone. People are sleeping.
Please, there's coffee at the back there, so.
Yeah. So I knew this was going to happen. I knew we were going to go for lunch, and I knew we were going to come back, and everybody would pay attention to Dr. Ebo's talk, and by the time it gets to everybody will be sleeping. So I asked ChatGPT, how do we break the ice and get people away from sleeping? And ChatGPT decided that I should tell you guys to get out of the soil, come on the surface, and let's play a little bit, and talk about other things that are also connected to the topic that we have discussed all day. So I didn't make this. ChatGPT did this for me in 3 seconds. So anyway, my name is Mohamed Amani. I'm a lecturer at KNUST. I'm working mostly in the Department of Wildlife and Range Management. I teach courses in range science. And I also have some affiliation with the Future Ecosystem for Africa project. I'll tell you a bit about FIFA, but for now, I want you to also enjoy my nice view from Kojai Street Nature Reserve. You've heard a lot of things about Dr. Ebo talking about LoRa. That is too far away from here. But when you move away a little bit from Kumasi towards where Dr. Ebo collected his data, you have to go through what we call the Forest Savanna Transition Zone, and you see all this beautiful landscape And this is quite often amazing, and this is the system that I like to study. You have to grab me here, or else I'll talk about forest and savanna until you are all asleep. So for my talk, I have sat through the presentations from this workshop for the past, I think, 3 days now. Yesterday I was missing in action. And I have heard numerous conversations about soil carbon and how we can improve soil carbon and how that can benefit climate change and other things. And I wanted to bring you guys, because when you meet a bunch of soil scientists, what do you talk about? Soil? No. I wanted to bring you guys a little bit out of the soil conversation, and let's look a bit more at the ecosystem level. So I'm going to show you some few issues that throughout my work with some organizations I've come to in terms of natural climate solutions, which of course all the things that we are talking about are part of. I wanted to emphasize that I originally trained as a forester, but now I work in Savannah. Why? Because actually, is there any forester here? No, I can say it. Because actually, Ghana is probably, you can say, it's a grassy or savanna country. And it has these unbelievably nice savanna landscapes. Imagine from the coast, so the coastal part of Ghana is a coastal savanna, actually Accra. Most of you came through Accra. Accra is predominantly a coastal savanna. And then when you come to Kumasi, you meet the forest a little bit. But if you go to north, then you find the Guinea savanna and all kinds of amazing savanna. And I'm showing you a bit of pictures here. Our savannas are open. They don't have a lot of trees. So you know what happens? People want to put their climate interventions there. Often people want to plant trees. People want to stop us from burning. People want to say that we can increase the soil carbon by changing how we graze. And there are a lot of controversies about this in the literature. So I wanted to bring some of this to your attention, as you saw on the first day when we started talking about trade-offs and all those. Most of the work that I'm presenting today is coming from a project that I did for the Future Ecosystem for Africa program, which is a program that is supported by the Oppenheimers in South Africa. And what we are trying to do in that project, essentially, is to see how we can enhance African voices in decision-making about transformative change, particularly in the climate change arena. Why does that matter? Well, Africa doesn't produce a lot of greenhouse gases compared to other continents, but Africa has something that other continents do not have. So let me brag a little bit about Africa now. We have the second largest land mass, so there's a lot of land here. We have about 60% of the world's arable land as terrace. It doesn't mean that all of them are productive. We also have very diverse ecosystems and biodiversity. In fact, Africa is the only continent that still has an intact of its full range of fauna, right? And of course, we have substantial opportunity for renewable energy. Basically, all this goes to say that Africa has a huge potential, in spite of Africa contributing very little to global greenhouse gas emission. We have huge potentials to actually play a particularly important role in global discussion about climate change and natural climate solutions. It is needless to say that Africa is also predominantly a grassy continent, which means a lot of the land area in Africa can be considered as areas where we can implement a lot of natural climate solutions. Being a grassy continent, and although we contribute very little to global greenhouse gas emissions, our grasslands do contribute to emissions to a certain extent. And I have colleagues here who are from animal science and colleagues from soil who have already mentioned this, so I'm not going to go over them. But I wanted to highlight two particular processes or two particular agents that are very important in this landscape when it comes to greenhouse gas emission. Number one, burning. You know, we burn a lot. Africa burns. No wonder we are hot in all capacities. Number two, we have a lot of substantial number of about 40 million Africans are involved in grazing in terms of keeping livestock as our main source of livelihood. And that also contributes significantly to greenhouse gas emission. So it is needless to say that in this case, savannas generally become a very good place where we can implement climate interventions, both for mitigation and also for adaptation. We spent a lot of time on one particular type of climate intervention or natural climate solution called sustainable agriculture. However, there are a few more, and you can actually categorize them into three main pathways. We have interventions that aim to protect savanna and other landscapes, so we try to avoid conversion. We have interventions that are linked to the management of this ecosystem. I just realized I made one mistake here. I did not put sustainable agriculture under manage because we spent the past 3 days talking about concepts, all of which fall under sustainable agriculture. But there are other concepts under the management regime. In fact, one of them, which has been touched a little bit, is the promotion or the proposal that for Africa to contribute to global climate solutions, we have to change our fire practices. Then there's also the aspect that comes from grazing. Under the restoration pathway, there's one particularly controversial intervention, which is afforestation. Let's plant trees. This is a big push. There's a lot of money for this, and there are a lot of companies in Ghana, for example, that are planting trees in savanna landscapes. I don't want to be antagonistic, so I'll just move on. So what are these interventions? For our project, we are interested in noting these interventions, and we're interested in also checking what is the evidence base. Like Dr. Yabua just showed, there's evidence, there's data. This is what happens when you have this amount of, when the soil pH is like this and the farm in Laura, this is how much carbon you are going to be able to store. We are interested in that. We want to find where the scientific evidence exists on the different interventions that are proposed for Africa. I'll run through some of this quickly, just highlighting only the key points that I want people to take away. So when it comes to fire management, the proposal is very simple. The idea is that we can reduce greenhouse gas emission or increase the carbon stocks in our savanna landscapes by, number one, changing the fire behavior in terms of how frequent we burn or when we burn, if we should burn at all, right? And the idea is that, obviously, sea inputs through biomass into soils obviously will increase soil organic carbon. If you burn less trees will die, and because of that, you have a higher above ground biomass. So it's all good. It ticks all the boxes. Tick, tick, tick. Everything works. We will see. The second level of intervention is grazing management. Here, we've already mentioned some of the grazing management concepts, so I'm not going to repeat them. But there are two concepts I realize people don't talk about. The idea of holistic management, which has been promoted by, I have forgotten his name, but he used to be a Zimbabwe farmer who now lives in the US and has a lot of money, who proposed that, actually, we can do a lot of good by adopting intense, short rotational grazing. You don't know the African farmer. We don't have time for that. But that is a proposal, and there are a lot of advantages that are associated with that. And then there's also the concept of the wilder rangeland concept, under which we have this big idea of rewilding. Let's bring back the wild animals and mix them with our livestock. And by having a mix of animals with different functional attributes, we can actually improve our lands, and we can actually improve how much carbon we store in this landscape. I just have one question for you guys. Do you think this is possible in Ghana? Anybody who says it's possible doesn't eat bushmeat. Right. So that is for grazing. For restoration, the idea is that our savannas are open. And it is true that they are open. They don't have closed canopy like forest. They are open because they have a lot of herbaceous layer, which allows us to also have a lot of things like our tourism, like all our amazing wildlife. What are they going to feed on, right? So, but the idea is that if you look at the African continent and where the different savanna types occur, there is enough rainfall. We can push a little bit more trees into these landscapes, and it should be possible to increase the carbon in the above ground biomass. This is doable, and this is what proponents push. You see, the problem is that the devil is actually in the details. Wait until we have to really do it, and then you see what the implications are. I knew we were going to be late and people were going to be tired, so I'm not going to go into all the details. I just wanted to argue that most of the debate and the contest in terms of the evidence, we have packed them in this technical report that we wrote a while ago. However, we have improved the report, and in recent years, we started publishing papers looking at some of these arguments that are suggested and trying to come out with a bit more stronger African perspective on some of these issues. For example, we recently published a paper that looked at the issue of reforestation, which included afforestation, and asked the simple question, what happens if you take off all the savanna landscapes, because they are savanna, they are not forests, so you cannot put your forest there. And what happens if you take all our croplands, because these are social safeguards. You cannot come and take the cropland and plant your trees. How much more area is left for people to really plant trees in terms of reforestation? And what do we see? Immediately you take all these considerations into the calculation, the amount of land that is available for reforestation drops by 90%. That is very significant. We have also recently analyzed the proposals for changing the fire dynamics in relation to try to increase soil carbon in the savanna landscapes, particularly with the proposal that we should move to early dry season burning because it has less emissions and it's less destructive. And we have argued against this, especially because African managers and African farmers have adopted different seasons of burning for different reasons. So using a blanket solution for this kind of policy directions doesn't really work for us. We've also recently published this paper, where it's not actually a paper, it's just like an opinion piece, trying to let the policy people understand that globally, climate solutions are not only for forests. Savannas are there, and savannas work in quite different ways, so we shouldn't push for just one agenda. Finally, I think last month, somebody is saying that battery is running low. My battery is also running low. Finally, last month, we published a paper where we said we have complained a lot about the different interventions that are given for African savannas and African landscape. So now let's try to tell people how to do the right thing. And I'm going to follow what my senior, Dr. Evoa, did by saying that please take a look at those papers because they touch on the issues and then we have discussed extensively there. I didn't want to bore you with the details of what we have written in the paper, so I decided to summarize the results for you. And I'll give you a few minutes to look at the results. In terms of the proposals that have been made for different interventions, and here, what we did was to look at over 400 papers in Africa, which looks at different interventions that have been implemented, and then we look at the evidence base. And what we have done is just a qualitative summary where we look at the benefit of every specific intervention from the case studies or the papers in terms of carbon, in terms of biodiversity conservation, and in terms of livelihood. And I must add that when we talk of carbon here, at that time, I'm not a soil scientist. So, and I got one soil scientist, but he didn't bring it to my attention. Here, the carbon includes both the above ground carbon and the soil carbon. So here, we look at carbon in terms of the total ecosystem carbon. And when you see a plus sign, it means that there is enough evidence that this particular intervention will yield positive results. When you see a minus sign, it means that this particular intervention is likely to yield negative or have trade-off or negative consequence on the thematic area that we are considering. And when you find the question mark, it means that the literature is not quite clear on the impact of the intervention. I must say that before you did this, I didn't know you guys, so I have not seen your papers. So take your papers out of it before you attack me on this. And I think what you'll find is that for most of the interventions, in terms of Africa, we actually don't have the science or the evidence base to speak to them. So it looks like people are more excited about the ideas and they push them vigorously before we even start to think about gathering the information or the data to prove whether it is correct or not. And in reality, when it comes to savanna landscapes and all these interventions that are proposed, we tend to find that for the ones that we have enough information on, the consequences are likely to be negative rather than what? Positive, right? So it calls for caution, especially when we are trying to deploy all these large-scale interventions that we are pushing for. So to summarize all that I have shown you so far, and of course some of them I didn't show you, but I'll still summarize it. I think it is clear, and everybody in this room will agree that climate change will have significant impact on Africa. The evidence are already felt, so across different sectors of the African economy, so we don't have to debate that. Natural climate solutions involving conservation of ecosystem, management of ecosystem, and restoration of ecosystem have a lot of potential to actually mitigate and also help us adapt to climate change. And Africa has huge resources that we can actually employ and deploy for global benefit. However, we have to be very careful because most of our ecosystems are grassy. They depend on fire, they depend on herbivore, they depend on some form of disturbance to be able to maintain them. And most of the climate actions that are pushed, especially those in relation to fire, large-scale grazing, the evidence seems to be very limited, so we have to tread cautiously. For afforestation, we don't even have to tread cautiously, we just have to cancel it out, right? And in fact, the hope for every proponent of every project is that we have a win-win, win-win for carbon and win-win for local communities. But actually what we have realized is that most of the time, the win-win doesn't exist, and rather significant trade-offs exist, and we have to be careful about that. But I am not the expert, so I asked the expert if they agree with my conclusion, and this is what they proposed, and it seems like they have a lot of confidence in the interventions, and they think it is working and everything is going forward. Psych, it is not true. I just asked ChatGPT, and ChatGPT produced this result for me. And then I asked ChatGPT to give me the sources of this result, and ChatGPT gave me the same papers, some of which I am an author on, a co-author on. So, two things. Don't trust ChatGPT, or don't trust the way I ask questions to ChatGPT. I was trying to conclude by bringing in a little bit of the project and what I have heard. I think you guys are doing an amazing job, particularly in relation to the soil carbon aspect, and the fact that we are considering climate, and the fact that we are looking at also potential trade-offs is particularly important. I think for savanna landscape, one of the things we have to consider is not only the soil carbon, but also the above-ground processes. And some of you have touched on that already. So still, I'll re-emphasize, when you look at the savanna landscapes of Africa, we have fire. Please study the soil organic carbon in relation to fire behavior and different fire practices. Let's look at the impact of different levels of herbivory, both livestock and wild animals, because they exist in our landscape. And let's look at the different types of ecosystem. The African savanna is not only about savanna. We have some forest patches in there. Not a lot, just small, right, that we all have to manage. On my part, I didn't mention this, but I run a small lab called the Plant Ecology and Nature Conservation Lab with these amazing guys. Some of them have graduated and left me. Some of them are still there, still doing slavery a little bit. And what we have done so far, we focus on Ghana's savanna. And all these pictures are from Ghana, so if you think I took it elsewhere, it means you have to come to my lab, so I'll take you to the field, and you can see all this nice view. What we've done so far has been to follow something similar to what Doc has done, to try to build a database and the data set for understanding the savanna ecosystem of Ghana. We have also undertaken a large synthesis, and actually what is funny is that when Caleb asked me to come for this workshop, he didn't know I needed the information, because the synthesis that we are doing, part of it is to look at about the carbon issues and how many papers we have in Ghana that have studied carbon issues in the savanna landscape. And I actually needed to come here to understand some of the mechanisms that you guys were explaining. Oh, some things have changed here. I don't know why. Anyway, we have also moved away from just the science, and we've started collaboration with the stakeholders in the savanna landscape to kind of try to bridge the science policy gap. And last but not the least, through our collaboration with the Future Ecosystems for Africa project, we also contribute to this kind of discussion at the continental level. And we have worked with Agnes, which is the, Agnes is, what is Agnes? Agnes is the group of expert negotiators who it's a think tank that help African government to negotiate climate deals. And part of the work that we do is also to feed information to Agnes so they can position themselves properly for African negotiators. Currently, we are running a couple of projects. Needless to say, not all of them are interesting to you, but one of them is at least interesting to you. In savanna landscapes, the herb layer is often the most dominant in terms of both the biomass and also the distribution. And we have a small project we have started that we are looking at how much the herb biomass contributes to soil organic carbon and carbon sequestration, and the dynamics of this as you move from different land uses and from different ecosystems in terms of the climate also. So we are doing a small study on this. And we have recently initiated a collaboration with our friend Young Joe, who is in UC Santa Barbara, and we just submitted a proposal under him to try to study soil carbon dynamics across the forest savanna boundary of West Africa. We're not only interested in just studying this, but we're also interested in trying to, just like you guys are doing, also to train the next generation of ecologists, especially African ecologists, people who will speak the language of the land and also at the same time speak the language of science and to advance development in Africa. I'm so open to more collaborations, so if you have something going and you're interested in working with us, please let us know. I wanted to leave you with this nice landscape. It's called Dija. It's actually a savanna landscape, which is a little bit close to the, not a little bit, it is surrounded by the Water Lake. It's one of the most unbelievable savanna landscape you have. Next time you guys are coming to Ghana, please plan to go and see Dija, and you'll see our real savanna landscape, and then you understand what the savanna in Ghana and West Africa looks like. Thank you very much.
Oh, okay. Thank you very much for the wonderful presentation, Amani. Do we have questions from the audience?
Mine is not a comment, it's not a question, it's a comment. I just really want to congratulate the teams. I saw when Dr. Yeboah was presenting and when Dr. Ahmad was presenting that you guys have published in very high impact factor journals. I was just checking the impact factor for nature sustainability. It's 32 and nature communications is 17. Congratulations so much on that. It's really encouraging.
Thanks.
Yeah, I'm interested to know in terms of the sustainability indices that you were comparing, whether this will be feasible, the plus, the minus. How did you arrive at this? Yeah. I'm interested to understand if you can show one of the slides where you have the management and then the protection, the grazing, the restoration.
It's on my screen, but it's not up there.
Oh, okay. But yeah, if you can speak to, I think it's the earlier, yes.
Yeah, I knew you were going to ask that.
Well, how did you know?
I knew you were going to ask that. So, look, actually, that's a good question, and that is a very fair one to ask. So what we did, we had, this was given to me as a small project by my collaborators in South Africa. So we had a very limited time to do this. And what we did was basically qualitative. We had to read the papers and then check, do they report a positive outcome of the intervention, or do they report a negative outcome, or did the paper actually touch on any of the sub-themes that we are interested in terms of the carbon contribution, in terms of most of the papers, and I think this is also coming from the fact that most of the interventions that are implemented in Africa are implemented by non-governmental organizations. So often the monitoring protocol that they design is usually not looking at the scientific angle of things. So the few publications that we have, we did more qualitative. And also, it is partly because a lot of these publications that we have did not really produce a quantified, I don't want to say things that will get me in trouble. But look, if you pick, for example, if you do a synthesis on Ghana and you look at the kind of papers that will come out, right, that have touched on the subject, and you look at what is in those papers, I shouldn't say it if you're for so we didn't have really quantitative measures from a lot of the papers. And that is why we basically did, does it talk about positive outcome, or does it talk about negative outcome, or there is no measure of the impact at all. And then in the end, we just summed it up. If you count all the positive and all the negatives, what does it sum up to? So that's what we use for this one. But in the subsequent papers, we have actually given more quantification of those.
Amani, a follow-up to Dr. Yeboah. Then you missed a lot of our forest island papers and our savanna ones.
So we actually, this one I showed only the savanna, right? But when we did it for all the biomes of the world. Once again, I have to be honest, we had a very short time, so we did only 400 papers across. And then in those 400 papers, we have to make sure that we included all sub-regions of Africa. So East Africa, West Africa, Central Africa, and Southern Africa. Even in North Africa, we included a few papers. We have to include papers from different ecosystems, forest, savanna. We also have to include different land use. So by the time you finish, the number of papers per each matrix is just about 15 or 20. But that is how much you could do in that period.
Amani, thank you for this wonderful presentation. I don't know if you included the Accra Plains as part of the, their beautiful nature of the landscape that we see, and in terms of climate change and ecosystem services.
I have to think. When we selected the papers, we have locations of the papers actually. And by the way.
Then you didn't see my paper.
I think one of your, from your group. Let me clarify this. All of them are coming from my former lab. So that's why all these questions. We are all associated to the same group of professors. But we did include papers from, the coastal savanna, because it is unique and only unique to West Africa in the way it works. So we included a few papers from the coastal savanna. I can't say it offhand if we included your paper. Yeah. However, my postgraduate student, Roxanne, sitting at the back there, now is actually, we are working to update the database that we have. We have moved from 400 to how many now? 1,700 and something papers. And as we are sitting at the back, we are trying to put the coordinates of where each of the paper comes from, and we are building the metadata with that. So I think hopefully before the end of the year, I can't, but maybe in the second quarter of next year, probably it will be ready already, and then we will try to share the data with everybody.
Please, any other question? Okay, Amani, let me go. From data we've been working for over the decade, one thing we are noticing is when you protect a savanna, wood tends to thicken, and soil organic matter also builds up. So the question I'm asking is, if you leave nature to its own this one, I think soil organic carbon and tree carbon are going to increase. What do you think?
I told you, before I answer the question, I'll tell the truth because you are trying to scheme. So this man is asking me a question that he knows where I'm going to argue from. The question has both a philosophical background, and then there is the scientific and practical aspect of it. So he started by saying that if we protect savanna, what does it mean to protect savanna? If you leave nature alone, what does it mean to leave nature alone? Do you mean to exclude fire?
No, fire
Or
included.
do you mean to exclude herbivores?
Everything nature apart from man.
But fire is nature. Fire is part of the nature of savanna ecosystem. So you cannot take fire out of savannas. It is just like taking trees from forests so that they don't have a closed canopy. And then you say that, no, that is fine. It doesn't work.
Oh, maybe, Amani, what I mean is if you take only human beings out,
Yeah,
what
but
we are seeing is savanna trees are thickening.
Yeah, for now, if you do, maybe that's what you'll see, but you have to understand, until human had a lot of influence, especially in Africa, you know who was the biggest ecosystem engineer? The elephant. If you take humans out, the elephant will come. And what do the elephant do? Kick everybody out, right? So, you are right to say that there's a lot of evidence from West Africa that suggests that if you remove fire and if you remove herbivore, you tend to have a transition to an often thicket kind of formation. However, what I have also realized is actually that there's a lot of conformational bias. So the experiments that were done in the central part of coastal savanna of Ghana, that did not lead to transition are not reported. And you can find them in the 1962 papers. They are there. The experiments were there. They were never reported in terms of their results, right? And then once again, you also have to look at the long-term dynamics, how the system used to work. Humans are not the only agent that cause fire in savanna. Natural fires from lightning, it's a very common occurrence in savanna landscape, right? Of course, humans have increased it. The other thing humans have done that we don't talk about is they have also fragmented the savanna so that the amount of burnt area in savanna, especially in Africa, has been decreasing since the 1980s. So we have to take all those nuanced approach into consideration when we are discussing this topic. To conclude, I'm a forester, so I understand the forest argument, but I have also become a savanna ecologist, so I have to always think of it in a much broader context.
Thank you very much, Amani, for the wonderful presentation. I think everybody is tired, but we have... Hello. I think we are already tired, so I'm going to be as brief as possible. Part of the work we were supposed to do was to also connect what we do here to what EJP arouNd is doing. I'm not going to dwell too much on the African dark earth here. A lot of the data which are supposed to be presented had been done by Emmanuel and Professor Luga. But what I'm going to tell, I intentionally wanted to do my presentation after Dr. Amane, because this one is going to be a political statement. When we look at the soil, I always say the soil is one of the most beautiful things when you go down to the microscope. These are all some of the most beautiful things you could see. And then Ghana has demonstrated that we can find this even in our soils with farmers using local nature-based solutions. I'm not going to present so much on the result, but one thing I'm saying here too is when I went further a little within last year just to check how healthy some of these soils are. And we are realizing that when you check almost all the heavy metals, they are all very low when you look at almost all the indicators. Unfortunately for us, when we look at, we don't really have very good standard for heavy metals in Ghana. But when you look at the USDA, I hope you get what I mean, environmental protection from other countries and the FAO, we are realizing that most of them are below. The only point we will have to highlight here is mercury. Mercury content is a little bit high in our African dark earth, and I'm suspecting that most of our soils in the past had been mined. So probably, I'm suspecting some of this mercury could be coming from that angle. Now, when we are talking about some of these nature-based solutions, increasing carbon and all that, what always comes into my mind is, if the farmer is not going to make enough money, then all the things we're going to do, we will say it nicely, give them money and incentives to do it, and after we are gone, they go back to zero. If they are not even getting enough, they go back to practicing small illegal scale mining, which we call galamsey in Ghana. So the question is, how do we, as a scientific community, come out with alternate livelihood solutions for these farmers? Because the argument we are trying to make with the INAS, which is the African Initiative for Nature-Based Solution, is that when farmers are okay in their pocket, then they will listen to us scientists. But so far as they are not, they will be engaged in some of these illegal activities. When I went to part of Ghana with some of our top researchers across the country, one thing we were noticing is you give them the intervention, they accept it, but in the evening, they go back to go and mine. And I met 3 ladies at a particular location, and we had an informal discussion. And the lady told me that she's able to make sometimes about 2,000 cedis a day, which is about $200. And you are talking, and she told me that you are telling us that you teach us how to do sowing, how to do farming, agroforestry. At the end of the month, I'm not going to get more than 3,000. So the question is, she just relayed to me and she asked me, do you think I'll go for your method or I'll go for the 2,000 cedis per day? That is 200 cedis a day. And I could not really answer. So part of our work, one of the things we are coming out with is, if a farmer is cleaning up the environment and storing carbon, then we believe that the farmer should be able to obtain carbon credit. Luckily for us, our governmental agencies have worked with a lot of international bodies, and they are already having some money stored in for them. But the problem we are having is, even though we are monitoring and measuring the carbon, our verification and reporting mechanisms are very poor. So early this year, I spoke to some team in Embrapa and then in the UK, and the idea was, why don't we come out with a monitoring device which can be able to do these 3 things in real time? Meaning that everybody within the chain responsible for getting the farmers credit will be able to see verified data backed by blockchain, meaning that me, the researcher, the farmer, or anybody within the chain will not be able to change or maybe alter the result. Luckily for me, the James Hutton Institute indicated that they've already developed a tool which they are calling the carbon estress. And all what we will need to do is to localize it in Ghana. Now, the problem we are having is, I'm not going to talk about the problems now because Dr. Yibo had done virtually everything there. So all what we are trying to do is everybody will be able to see our data stored in the cloud. But as Abad was saying yesterday, we are not going to trust artificial intelligence whole and whole, meaning that we are also going to do a lot of statistical modeling using our squares to be able to double verify whatever thing we are saying. Because sometimes the whole problem with artificial intelligence sometimes is, you've done the work, you've given the data, you get virtually almost 99% this, but when you change the scenario to other place, maybe if you use 30, normally what they do is they use 70% of the data as against 30% for verification. But the question I asked them is, why don't we use 20% and then we try the 80% for verification, and then there's problems. So the question is, can we also do some physical modeling? So in this particular work, what we're doing is we are bringing out 5 ladies, and each of them are going to look. Some are going to look at the gender, some are going to look at both the mathematical and then the AI solutions, and others are going to look at ecological indicators. And at the end of the day, we want to get a verified system with almost all stakeholders involved to be able to get farmers what they deserve. So the app we're going to use, which is going to control the whole interface, is called Retina. And almost all the benefits we've associated so far had been indicated by our AI team. Now, the second part of the issue was, now the lands are destroyed. So what are we going to do about the destroyed land? The alternative solution is afforestation, but as Amani is saying, even for carbon storage, the evidence now is low because most of the trees, most of these things came about 5 years ago, and we are not really seeing their benefit yet. But the question for the farmer is, they need some money in their pocket. Local communities need, but almost all their lands are spoiled. So we came out with a sustainable management initiative for livelihood enhancement, which we call the SMILE. And this time we are targeting only Ghana farm forest landscapes. I think I don't need to speak too much. So the body responsible for small-scale mining in the past was called the NILP, but it has changed into responsible mining currently. And the idea is we want to target communities, and we want a solution where the community is going to benefit directly. Now, most of the lands are polluted with almost all the heavy metals we talk about. So what we are telling them is we are not going to do anything food in that environment, but we are coming out with rubber plantation. So all what we need is to change the landscape to be able to support rubber plantation within that environment. The most critical resource for rubber plantation is water. So, and they don't like fire at all. So most of the work we're going to do will be around high rainforest zones across the country. And as I indicated, our targeted communities are always the community. Because if the communities are not benefiting directly, then almost all the intervention solutions we are talking about will not work. So in almost about 16 communities across the countries, what we're going to do is we are setting up 1 hectare plots where we do the rubber plantation. The soils we have down there nowadays, I call it no soil at all, because you have almost about 100 centimeters of the whole entire soils mix. So the question is, what is that? So part of the whole thing is then we will need to get topsoils. Luckily for us, we have a whole lot of mountainous areas left across most of the places we do. So the idea is you go for the mountainous soils and then fill the top. And then within about a year, we are we are planting the rubber plantations, and then we are monitoring some of these things in real time. Now, to be able to also do some of these things very well, we will have to look at if almost all the soils are polluted, then one way or the other, we will know that the water will also be polluted. So we are coming out with tools which will also do real-time monitoring of the heavy metals. And one of the tools we are almost finished developing is the Gallam Sense, which is able to monitor some of these metals in real time, meaning that, and then we have a lady under the Artificial Intelligence for Sustainable Development also looking at ways to clean the water. What it means is we are going to sieve a pipe, and she has developed a technology which is able to sieve the heavy metals out. Recently, she even indicated that her first lab experiment indicated that even some of the beneficial nutrients from the water itself were filtered out. So she is now looking at ways to be able to allow some of the beneficial, this one, to pass through her filters. To be able to go through some of this stuff, farmers need to plant across the country. But the thing is, they depend on the rain, and they sometimes depend a lot on the meteorological agencies, who are not close to them. Most of them are in Accra. So the question is, how then do we solve their problem for them? For us to talk about carbon sequestration, then the farmers must be able to plant at the right time and perform the right management practices to be able to get the yield they require. So we came out with the Smart Indigenous Weather app. The idea, I'm skipping, I know we are already late, so I'm just trying to do so. Now, farmers in local communities look at certain ecological indicators to be able to predict rain. And part of our analysis recently is indicating that, let me get there first, it's indicated that farmers within a 24-hour period are able to predict rainfall, and the accuracy is almost around 82%. For the meteo in Ghana, they are almost ranging around 80 to 85. So the question now is, can we be able to merge a technology using the farmers' knowledge? Okay, so one of our good professors, who is an agric engineer, had worked a lot with some of these ecological indicators, and I'll call Dr. Enoch Bessa. He's sitting at the corner there, came out with very good weighted, and he's even trying to encourage pious to be able to weight some of these ecological data so that we include artificial intelligence. The whole idea is farmers are doing something which is able to predict nature, but the question is, how do we use innovative approaches to be able to improve their technology and then their accuracy in predicting weather? So what they are looking at is heat. Sometimes they look at the formations in the star, they look at cloud formation, they look at even the sun, how warm they are feeling. I remember that is not here. A farmer told me that when he sees, bamboo, flowers of bamboos, he knows very well that within a week or two is going to rain, or the major rain is going to start. And it works for him most of the time. So they look at the moon, the wind, the fog, and then the dew. So all what we're doing in the last 3 months is to collect a lot of data where Enoch and Pius will be able to wait and then come out with very good models. So that and almost the app is already built. It is on farmer fields, and the farmers are working with this seriously. In all this, we're talking about Africa, but some work we've done in Europe is almost showing the same trend Dr. Yeboah showed. In a nutshell, I told my team members in Kosovo and Hungary recently that we will now classify Kosovo as an African soil. Upon all the statistical data we've done a whole lot of modeling. But one thing we are realizing in Kosovo right now is soil characterization, that is, soil type is the most important parameter when you are looking at carbon storage, including nitrogen and then sometimes phosphorus. Phosphorus is a little dicey because elevation and those kind of things all comes to affect its drivers and how they influence distributions across most of Kosovo. But when you come to the land uses, the forests are still having very high, I cannot say high, they are having higher soil organic carbon. And most of the soils we studied in Kosovo were having soil carbon less than 2%. This is supposed to give you an idea. The soil types are increasing across a precipitation gradient, and you realize that when the soil organic carbon and almost all the nutrients are going and tending across some of these layers. So the idea now is, first, when you are going to a field, for we agriculturists, we know that soil type will always give you a limitation. So I did not really want to worry Amani too much when he was doing the work, because part of my question was, did you consider the soil type you are dealing with and how they will contribute to your count? So in some of these collaborations, I think we may need to talk more to see how we can be able to come. Not only in Kosovo, but the same similar thing we observed in Kosovo was also observed in Tanzania under the Horizon 2020 project, that is Soil for Africa. And we are realizing that soil organic carbon is almost also tended to, when you do the modeling, you are sometimes getting about 82%. You are getting, if you look at the R square, but sometimes you realize that majority of the whole variability, about 60, 70%, is only by soil type. So we are just trying to give you an indication that within our system, including some parts in Europe, soil characterization is important. You just don't need to get to the field pick your data and come and do your analysis, but you must know the type of soil you're dealing with. And then I think I'll skip too much. But in all the interventions you are dealing, in Kenya, we came out with a clear indication. Much of the farmers are telling us that about 97%, almost 100% of farmers are telling us that when you do regenerative practices, you are going to increase your yield. And that is a good indicator. They are also telling us that they are able to control, about 60% are able to control their erosion using the practices we indicate. And then they are also telling us that the soil gets darker and soil structure is improved. But when it comes to climate change, as we all say, it is something they cannot really measure, so they find it very difficult to talk about. In a nutshell, in the past 2 or 3 years, and that is one of the main reasons why I came to Ghana last year, we are dealing with rubber establishment. And luckily, I was made the rubber researcher for the rubber industry in Ghana just about 2 months ago. Part of the work we want to do right now is look at how we can be able to improve things which can be able to improve the livelihood of farmers. And we believe that with rubber, the only thing you need to do is to take very good care of your plantation within the first 2 years. If you are able to do that kind of stuff, then after 5 years, you can be able to harvest this commodity for the next 50 years if you do proper harvesting. So part of the work we are doing, led by Dr. Erasmus Tete, is to look at how to find 5 climate-resilient clones which are specified to specific areas across the country. So far, we are dealing with about 4 agroecological zones, which are almost all tailored toward the rainfall areas. We are looking at virtually all the things soil science look at, but we are now going into the innovation. We are going to look at moisture stress, and some of you here, we are inviting your knowledge to come and help us in this scheme. It is very young, and we will start going to the field from next year. And almost all things which soil researchers do, we know. I know Abad had already shared some paper, arouNd, which relates to some work which has been done on rubber farms in France. So we'll be looking at some of this stuff and then see how we can include our AI team in Deeper Lab to be able to monitor some of these processes in real time. And the idea is, at the end of the day, if we improve the life of farmers directly, and then we are now telling them about carbon storage, food security, and all the other regenerative practices we want them to include, they will be able to do so. So in a nutshell, what we want from scientists today is good quality data, which I'm telling data, but they should be good quality. I know Amani was unable to say some of the things directly because the thing is sometimes you get the paper, but the question is, do you trust the source of the data? Do you trust the quality of the data? And will you want to include that data in your modeling? And we believe that when we get quality data, that is why sometimes I'm now moving to the IoT-enhanced blockchain approaches, because with those data, nobody will manipulate. And if we are able to increase our measuring scheme, then we can be able to build better local climate agriculture scenarios for better food security across Africa. Thank you.
Doc, one question. You mentioned that one of the ways you are going to use to maintain or to manage the galamsey is to gather topsoils from mountainous side to the place. Is it not like robbing Peter to pay Paul in a way? Because we know as soil scientists that when a soil is disturbed, emission also in a way goes high. And we are now going to handle a problem. Do we have to go and gather the topsoil from a mountain to come and take care of that?
to be frank, I love your question. So part of the whole thing is after disturbing the mountains, we try to grass it, and then we try to restructure it with our engineers. The whole idea now is we are at a dying situation. Most of our land, especially around farming communities, are almost all gone. Most of them are mining. And the question now is, are you going to give them alternatives? Something? Because almost all farmers across the high rainforest believe in rubber plantation. They believe in the tree crops in general. But you can't plant coconut or those kind of things down there. And then the soils they are having in their communities now is no soil at all. The apparent material. So the question is, how do you go it? So sometimes I believe that we have to go through the hard way first to improve their livelihood, or for them to be sure that probably in the next 5 years their livelihoods will be assured. So that everything, and all these things we are doing, majority of the fund, we are dealing with banks and trust system, so that almost all the funds goes to those trusts, and the trust will distribute it to all the interested parties who owns the particular.
Looking at, recently, I'm a senior man where Sefi Bekwai, yes. And along the way, looking at the number of hectares that has been degraded as a result of Galamsey, where can we get this number or quantity of soil, topsoil to be specific, to handle this matter? And also, did you do any kind of analysis to know that, okay, this is a hectare we are having and this is the soil we are bringing. Okay, where we are bringing the soil from, what is the problem we are going to create that later we are going to solve? Weighing the two, have we checked all those balances to know?
So I'm going to show you this. In the Galamsey areas, even what we call the post-Galamsey area is still a Galamsey area. As Enoch will tell you, when you leave the farmers, you know, after the first Galamseyers led, I'm sorry to say, especially by the Chinese go, when they leave, the local communities are also having this new amalgamation method, which they continuously mine. So the question now is, if you don't do something drastic to be able to solve their problem, then they are going to continuously destroy the soil. I hope you are getting where we are coming. And the question you asked, are we going to get that large number of soil to be able to do this kind of stuff? I believe no. But where we can be able to do some of these approaches, we do, and we are not only doing rubber plantation, we are calling it livelihood. So honey, bees, and all other things. So what we are going to do is, even in a cocoa farm, the question now will be, should you only be able to get money from your cocoa farm? We believe that teaching you methodology for mushrooms, beekeeping, and other things which can give you alternate sources of money can be able to solve their problem. But we believe that in the degraded areas which are mined, the rubber could be one of our main solutions. Okay, we are done. Oh, okay, so I think we've done a whole lot for the whole day, and we are done, and our visitors need to go home to go and relax, because we will need to pick them up again.
Yeah, but I think there are, if we still have our online participants, and then if Melissa is still around, maybe just a few words from you, and then we can adjourn the meeting. And tomorrow we are likely to miss one or two. So Mabad will be leaving us. Unfortunately, he would have to go back. But we will share the virtual assessment form to you online so that you can give us a good assessment of what has happened. So, Melissa, if you have some few concluding remarks for us, and we also want to hear from Abad before he leaves, finally. Right.
Okay, thank you. Yes, today I think was very, very exciting because unfortunately, I lost the when the meeting starting, but I assisted lots of students presenting, and I love it. I love the youngest researcher presenting your own work. I think it's very, very interesting. And This presentation about the Ghana reality, how the farmers are living, or the problems they are, the concern the farmers. I think it's very open my mind, because it's very, very similar that the problems that we are actually we have in Brazil. Here at Embrapa Rice and Beans, we are living in the Cerrado. So it's kind of similar to savanna, including, but we don't have a dark earth here, just in the Amazonia. But here we have a climate, the kind of vegetation is kind of similar. And we know that some public policies could help to farmers to establish these technologies that we are seeing that it works. And these technologies brings not just lots of benefits to environment, but lots of benefit to the farmers, because they when they are using integrated systems as crop livestock or crop livestock forest, they not just improve carbon on soil, but they are diversifying the farm benefits, as sometimes they have wood to sell. Sometimes they have cattle or milk, and sometimes they have cash crop. So we can end the carbon on soil. So I think one of the things that we could think in our meeting, and not just how we could improve GHG, avoid, in fact, GHG emissions, but how we could adapt or deliver these techniques that farmers could use to adapt their environment, but how we could deliver some just more information, or try to help our colleagues to establish some public policies to bring it new technical publications or something like that, to for example, using some from Brazil that we have similar conditions, and maybe we could think on it. If, of course, if it was the Ghanaian people and the colleagues from there are interested, of course. But thinking on event, I'm very very happy with that, and I'm very sad to lose the event for tomorrow. I would like to yes, I would like to go there to visit the field, to eat the picnic, because I'm thinking that you have lots of delicious food. But I'm here, and when you come back on Friday, we can have joint discussions again. So, and I would like to say thank you for entire team to provide all those delightful actions, not just from scientific actions, but cultural actions too, and this kind of interactions between the farmers. I think this is very important, because seeing the real life, we can apply the technology in that. So I think this is more important for us as a scientists. And I would like to say thank you to Abad too, as a deputy coordinator of this project. He is representing us and put all the information, bring some information for me and help me to write things too. He is sent some emails for me, and I would like to say thank you very much for all of you. Thank you.
Yeah, we are grateful to you too. Thank you too for sacrificing all this while for us, giving us the direction, and then setting us on the right roadmap. In Ghana, we normally say that when the king speaks, that will be the end of it all. But for now, I will break that protocol and I will allow Abad to say a few words because he's leaving us tomorrow. So Abad, just a few remarks and then you say bye-bye to us. Excellent.
Please use the microphone, Abad, because otherwise I can't hear you. Please, please.
Yeah, just I said that Edward told me that a few remarks. I said that you said the most important thing, so I have not too much to add, and I think this is another day which is full of information, but it was an exciting day because we discovered the work that the students, the PhD student doing, and yeah, of course, this is some advancement that I can see here, but it's, of course, there is improvement. This is all the time. We need improvement, the way they present, the way they select the result. But also, I think that is the diversity of the information that we see today, with covering different topics, specifically the ones that dealing with this continent, but also others. And yeah, it was really, for me, it was 3 days now. It is full of full of energy, and yeah, I'm really also sad that not to be participating tomorrow for the excursion, because I have to leave. I have a PhD student on Friday afternoon, so I cannot miss that. Otherwise, I will be in trouble. So anyway, it was a very good, and I really enjoyed also the last presentation of Caleb, which was with energetic presentation and the way how we are dealing with the farmer. And of course, you know, the farmers is always something tricky, and then but also it hangs to the policy. And I can share also some of the experience from what I see in Morocco, for example, that they have some exotic plant imposed to the farmer, for example, avocado in an area where there's no water, and then just because some people taking benefit from this plantation and then giving some sand to the farmer, so they are always continue to be in trouble and poverty, but the other, they can make money. And there's also some sustainability issue behind these things, not only for respect to the water, but also for the system itself to maintain it. So not only destroying the system, but also making more and more poor those population that are living from this agriculture. So this is, I think this is, since I'm in the university, I've been hearing about these things, and then always the farmers are paying the price. And then if you see worldwide, regardless where we are, in Europe, and I don't know in New Zealand, but in Europe, in Africa, in North Africa, is always they are suffering. It is really, there is no improvement, and they are doing the big job. But something is going wrong with what we are doing, and something is also for the policy that they are not probably taking the right things to do and to improve their living standard, but also the system itself, because we need the farmers and then we need the system, so otherwise our existence will be not anymore make sense. So I think that this workshop and the topic of this round, this was a very, very good success, and I hope that we can give some of the examples for other. I think this is, I have been in quite say around the EJP project, but not this one, which is really not because I am just sending some flowers, but it's really a diversity, and it's quite different from what I see so far. And yeah, congratulations for this hard work and for Caleb. And I was close because the administrations, I'm getting old also, and I don't have no passion also for papers and visa and all of this, and I have been bored in Caleb. That's what is going on. But I don't regret to be here and then discovering these things. Yeah. Thank you for all, and thank you for the student. And really, we keep in touch. I think that we are going to have another year extension of the project. Did you hear, Melissa? I received the report this morning. And then I can share with our funding agency, and then everybody would be happy. Yeah. So thank you very much. And then...
Right. So thank you very much, Sabat, for the reflections. We are excited about this. So on this note, on behalf of the local organizing committee, we would like to say thank you very much. We want to thank our online participants. We also want to give thanks to the IT team, those who have been working very hard to keep us going, and to those who have been providing the catering services for us. We appreciate it very much. The meeting is adjourned until tomorrow, 5 a.m. for those traveling to the field, and those that are traveling back, we say bon voyage. The meeting is adjourned. Thank you very much.